So hello students, welcome to Vedantu NEET English. This is Basuraj sir, your biology master teacher. Welcome to the one-shot lecture of biotechnology. Now all of you are aware there are two chapters. This is the first chapter and then we have the second chapter, right? The first chapter, principles and process, will be taken by me, as well as the second chapter will be taken up by Gopika ma'am, right? Now students, before that, quickly let me know in the chat right now if I'm perfectly audible and visible to all of you. By thumbs up in the chat or fire emoji, heart emoji, or whatever you want. Quickly let me know in the chat right now if I'm audible and visible to all of you. Quickly let me know in the chat. All of you. Quickly let me know in the chat. Yes, am I audible and visible to all of you? Yes. Now students, with that being said, can I ask you all of you a very simple question? Usually every single class of mine starts with a question, right? The question is very simple. The question is, when you hear the word biotechnology, I repeat again, when you hear the word biotechnology, tell me what exactly comes to your mind. Do you think biotechnology is a very old process, or is it a very new process? Tell me students, quickly, let me know in the chat. What comes to your mind? Like biotechnology, is it a very old process or is it a new fancy process? You know, last year it was formed, very new things happen here. Tell me what comes to your mind. Now, if you think it is a very new thing, let me tell you right now, you are wrong a little bit there. Why? Because biotechnology, right? Biotechnology is a concept. It is actually very ancient. It is actually very ancient. For example, how many of you love eating curd in the house? Tell me. How many of you love eating the curd in the house? I love curd, right? Making the curd is also biotechnology. How many of you love dosa? Raise your hands. Dosa. Making dosa or dosa is also biotechnology. So what exactly is biotechnology, students? Then what exactly is biotechnology? Biotechnology is very simple. What is the meaning of bio, students? Tell me in the chat, all of you. What is the meaning of bio? The meaning of bio is living. The meaning of bio means living. Bio means living, and technology means a process or a technique. You know, a technique is used. So we are technically using, right? We're technically using living organisms. It could be a cell, a tissue, a process. It could be any microorganism. We're using them to make products. That is called as biotechnology. That is called as biotechnology. So if you want to make bread in the house, use yeast. So you're using yeast to make the bread. That is also biotechnology. You're using living organism. You're using a technique for human benefit. That is biotechnology. Now students, let me tell you, if you have zero idea about this chapter, let me tell you again, if you have zero idea about this chapter, you do not know what is a plasmid, you do not know what is a cloning vector, you do not know what is a PCR, what is recombinant DNA technology, do not worry. Because let me tell you, it is my guarantee from my side to you. If you attend the lecture till the very end, I'll repeat again, if you attend the lecture till the very end, every single concept in this chapter will be clear to you, and at the same time, you will be able to solve every single question. Now the question reminds me, students, do you think how many marks this chapter is? Anyone, any idea how many marks this chapter is? 44 marks. Last year, NEET 2024, there were 8 questions from this chapter alone. So this is a very high weightage chapter, so I would advise all of you to completely lock in. No distractions at all. Don't look, no looking left, no looking right. Completely fix to my words. Listen to my words. Follow through. Keep your NCERT next to you, and let's start. Let's start the class, students. Let's start the class. Now, students, I told you biotechnology is very old process. Yes, biotechnology is a very old process. Now, one question arises. One question arises to all of you, right? The need. Like, why do we need it? Like, why do we need biotechnology? Anyone, any answer? Why do you think we even need biotechnology? Like, is there any need for it? The answer is very simple, yes. Now, I'll give you a very simple example. Listen to me very carefully, okay? Now, how do you send a message to your friends? Let's say, Meghana wants to send a message to a friend. How do you send messages? Either you will take your phone, Instagram, WhatsApp, Telegram, or normal message. Take your phone, type the message and send it. Done and dusted. Okay. Now, what about back in the days? How do they used to send messages? They used to write letters. How many of you remember? They used to write letters. They used to send pigeon. In Akbar Birbal time, they used to send people as messengers. They used to send people as messengers. So as the advancement happened, as the advancement in science happened, it became easier for our own life. It became easier for us to send a message now. All you need to do is take your phone, drop a message and send it off. Similarly, there is advancements in biotechnology. There is advancement in how we can use the organism better. Like how we became better at sending messages, we also became better at using things, using animals, using plants for our advantage. And that gave birth to the biotechnology. Okay. Now students, listen to me very carefully. This wine making, curd making, bread making, all of this comes under traditional biotechnology. Traditional means very old method, very old method. Okay. Now, since of science of using living organisms or the products for benefit of human and surroundings. So we are using animals for the benefit. Okay. Now this reminds me, students. This reminds me. How many of you have heard about selective breeding, students? How many of you heard about phone pay? Now phone pay also send messages, and I have seen, students. I don't know why. Out of all the places, you want to send even message on in even YouTube chat. Like I have noticed, like before the start of this class, there are 2-3 people texting in the YouTube chat also. Some weird way of communication, but okay. Everyone has their own ways of communication. Now students, what is selective breeding? I'll give you a simple example. Listen to me very carefully. Okay, simple example. Okay. Let's take a small mango here. Small mango. But this small mango is very sweet. Simple example I've given before also. Very sweet mango. Okay. Very sweet mango, but it is very small mango. Okay. Now here on the other side, let me take a large mango. Large mango, but it is not sweet at all. Not sweet. Now what do you do, students? What do? You take this plant, and you combine both of them and you get a new baby. So you end up getting a large mango, which is also very sweet. Yes, can we achieve that, students? Very large mango which is also very sweet. We can achieve. It is called as a hybrid here. Now students, let me tell you one small sad story here. The sad story is that are there any chances that if you are extremely unlucky, would you end up getting a small mango which is not sweet also, which is completely not sweet? Are there any chances of this product? There are chances. So this right here is the most unlucky person, and also at the same time, what exactly happened here is all the undesirable trait also came together. So in your traditional biotechnology, the chances of this happening is also there. So for the very same reason, then we came to the modern biotechnology, like all of you. Now students, how many of you remember COVID time? How many of you remember the COVID time? COVID time was very, very difficult and fun for few people. For example, all of you were in class 9th or 10th, right? Class 9th or 10th, you're like, oh, no board exam, you're chilling, playing PUBG all day, doing something, right? COVID time. In that COVID time, students, there were also a lot of superheroes working somewhere. Who are the superheroes? These are superheroes who are wearing a white lab coat and working in the laboratories, working in the hospital. It could be nurses and doctors, and also biotechnologists. Now, do you remember, students, how did this entire COVID came into halt, right? How did this entire COVID came into stopping of entire thing? This entire COVID came into a complete halt after the introduction of vaccine. How many of you remember the vaccines? We used to get one shot, second shot. Sometimes it was very expensive also. Sometimes we used to get it for free. There was a booster vaccine. Right? That was the story of vaccine. Students, because of this vaccine itself, the entire the COVID era came into an end. And let me tell you, students, if there was no biotechnology, there would be no vaccines. So who is the real hero again? Students, to all of you, who is the real hero? The real hero of your COVID time, one of the real hero was the biotechnology. So that is the importance of this particular chapter. It teaches you that how important this particular subject or this particular field is. So all of you in the chat, biotechnology OP in the chat, all of you. Biotechnology OP in the chat, because without biotechnology, we would still be stuck in the entire COVID time. A lot of people dying. For matter of fact, the first vaccine created by Edward Jenner cured a lot of people, right? Not only vaccines, even antibiotics. All of you keep up antibiotics, right? You fall sick, you take an antibiotic, you're all better. Now imagine a life without antibiotics. Imagine a life without antibiotics. Yes, there would be no superbugs, but you will also be in a very bad state, because these bacterial diseases will be very susceptible. So very bad life. So antibiotics, first antibiotics by Alexander Fleming. Yes. So all of these antibiotics, vaccines, all of this are because they are here with us in our surroundings, in our body, because of a field of biology called as modern biotechnology. That is techniques and process used for manipulation of genetic material. This genetic material, students, this genetic material could be what? This genetic material could be DNA or it could be RNA. You're manipulating genetic material of living organisms for our own benefit. Students, can you imagine a life right now without biotechnology? In this current era, it would be almost impossible. That is the importance of biotechnology on our life on a day-to-day basis, right? So traditional biotechnology, modern biotechnology, both desirable and undesirable genes are inherited. In modern biotechnology, only desirable genes are introduced. So this is your modern, this is your traditional biotechnology. Now students, this is a definition. There's a definition which all of you need to know. This is the definition you need to buy out also, unfortunately, because in biology, few things you have to remember. This definition is not given by me, not given by you, but this definition is given by your European Federation of Biotechnology, EFB. That is the integration of natural sciences, the growth and everything, and organism, cells, part therefore, and molecular analogues for products and services. So we are using organisms, using science, growing them, extracting their genetic material, that is the molecular analogues, extracting the DNA and RNA for products and services, that is making vaccine or making antibiotics. So I'll read this definition one more time. I want every single student watching this lecture right now or watching it recorded, I want everyone to read with me. Can we start? Yes, all of you. Now, integration of natural sciences and organisms, cells, part thereof, and molecular analogues for products and services. Okay, so that is a simple definition. That is a simple definition of biotechnology. So don't need to worry so much. Very easy chapter. Okay. Now students, if you have watched the video till now, I want everyone to quickly smash the like button, because Gopika ma'am has given me a target to make you 200 likes minimum before Gopika ma'am comes. Okay. So all of you keep up the target, smash that like button, all of you right now. Otherwise I will get one target on my head. Okay. Now students, let's go to principles now. Right. Principles of modern biotechnology. Principles. The entire biotechnology, right? Entire biotechnology can be divided into two separate categories. One is called as genetic engineering. Other one is called as bioprocess engineering. Now what exactly happens in genetic engineering? Listen to me, I will tell you. Very easy it is. Students, all of you know this is a DNA. This is a DNA, right? Students, can anyone tell me what is the smallest segment of a DNA? What do you call the smallest segment of a DNA? For example, here we have a gene. Here we have a gene. Okay. We are taking this gene and we are introducing into a host. Obviously, we need to cut the gene first of all from here, or we need to cut the piece of DNA. Cut. We are cutting it. Now, how do we cut it? We can cut it by using a molecular scissor, magical scissor, which I'll tell you later on. What is this scissor? What does scissor do? Everything I'll tell you later on. Okay. Now students, if you cut this particular piece of DNA and add it to a particular host, we can add it to a host. Now students, tell me in the chat, can I do this process of genetic engineering on the roadside? Is it possible? Can I sit on the roadside and do this entire genetic engineering? Not possible. Can I do this in open ground? Not possible. Can I do this in my classroom? Not possible. This entire process of genetic engineering needs to be done in a lab. For example, here I have a lab. Here I have a lab. In this lab, I have complete sterile condition. I have complete sterile condition. Complete sterile condition in this lab. So to maintain this sterile condition, the maintaining the sterile condition is called as bioprocess engineering. So what is bioprocess engineering? Bioprocess engineering is maintaining the sterile condition and only obtaining the desirable products, nothing extra. This entire process of cutting the DNA piece, taking the DNA piece, attaching somewhere, transformation, all that is part of genetic engineering. So all the hard work is in genetic engineering, maintaining the space sterile and making sure only desirable things are growing. That is part of bioprocess engineering. Clear? Clear all of you? Simple difference between both of them. Now, techniques to alter the genetic material of the organisms to change its genotype and phenotype, that is genetic engineering. Now students, let me tell you what is bioprocess engineering? Maintenance of sterile condition to enable the growth of only desirable microbes to obtain the pure products. That is bioprocess engineering. Now I believe till now there is no doubt. If there is no doubt till now students, let me tell you, the entire chapter is also this easy itself. This blue and white screening, entire chapter is very easy. Okay. Now students, can I tell you a small story? Now I will teach you the entire chapter with the help of a small story. Okay. For that I need two characters on the board. Okay. So I have two characters here. I have one person here. Okay. Let's say his name is Bro. Okay. We have one more person here. Let's make him in white. We have two people here. Okay. His name is Mucha. Okay. We have two people on the board. All of you are aware of them. Okay. Two people are here. Okay. Let's take, for example, Bro and Macha are the two people here. Okay. This Bro right here is around 6 feet tall. 6 feet tall. Okay. And Macha here is around 4 feet tall. He's around 4 feet tall. Okay. So this particular person, Bro, is around 6 feet tall. Macha is around 4 feet tall. Now Macha is like, bro, how did you become so tall? He's asking, like, how did you grow so tall? Is the comp plan, is it the boost, what it is? Let me tell you, nothing all that big time works. But what is the main secret of your energy? Like, why are you so tall? Bro, basically, Macha is asking Bro, right? Why are you so tall? But Bro is like, bro, I've always been tall. It is in my genes that my dad was tall, my mom is tall, I am also tall. So all the genetic information of Bro being 6 feet height is present in its DNA. Now what happens is, Macha is very jealous. He's like, what? I have to do something about it. I cannot stay quiet, I have to do something about it. Okay. Now what does he do is very simple. Listen to me very carefully. When Bro is sleeping in the night, Macha is going to his house. He is putting some chloroform in his face. He is fainting the entire Bro. And after that, from the Bro's skin cells, or he takes out one random cell from his body. Right? For example, this is the cell right here. This is the cell right here. Okay. This is the cell right here. Now students, in this cell, is there any DNA present? Yes, there is DNA present. For example, this is the DNA right here. This is the DNA right here. And this DNA right here, right, this DNA has all the information. This DNA has all the information of why Bro is so tall. Why Bro is so tall. All that information is given here. It is given here. Okay, listen to me very carefully now. Now what it does is, Macha is like, wait a minute, now I have a piece of DNA. I have cut the DNA and I have isolated this particular piece of DNA. Now this is the first step which is happening here. First step. Okay. What is the first step called as? First step is called as isolation of desirable gene. The gene for becoming tall was the desirable one. He went on and he took the genetic material from the bro's blood. He took it off. Okay. So first one is called as isolation of desirable gene is done. Isolation is done. Okay. Now students, listen to me very carefully. Next step, what is happening is he is taking this, right? Let's say this is the DNA. This is the DNA, right? This is the DNA, and here we have the desirable gene for, let's say, tallness. Now he is taking this and he is chopping it, the two different sides of this DNA. Let's say he'll cut here, he'll cut here. How is he able to cut, students? He is able to cut on the two sides because of a molecular scissor, which I'll tell you later on. Again, don't worry, everything will be taught to you. Now this particular segment is removed out now. This particular segment is removed out. He removed this segment. Okay, this segment right here is very special segment. It is responsible for all the tallness. Okay, now he is taking this particular piece of DNA, also called as alien DNA. It is also called as what? Alien DNA, and is transferring it into a vector. What is a vector? Don't worry, we will learn this entire thing later on. He will transfer it to a vector. Now students, this is a vector right here. This is a vector. This is a vector right here. To this vector, he is adding this piece of DNA. Let's add it here. Okay. To this vector, he added this entire piece of DNA. Now students, he is transferring this entire thing into the host. Who is the host here? The host is going to be macha here. So this vector is added to the host right now. Is added to the host. That is called as transformation. That is called as why is board not writing properly today? That is called as transformation. So he is taking this vector, adding into the host. That is called as transformation. So what do we learn? This is your entire chapter. Technically, this is your entire chapter only. Okay, listen to me very carefully. First, what is the first step? First step is called as isolation genetic material. Done. Isolation done from here. What is the second step? Second step is transformation actually. Now before transformation, you need to cut the DNA, molecular scissors, take the DNA, add it to a vector, and vector is transferred into the macha. That is called as transformation. Now after that, third step is what? Third step is to make sure there are multiple copies of gene of interest inside the host. That is the third step. Now, students, this is the entire framework of your entire chapter. Transformation, entire working into a vector, vector to the host, that is called as transformation. Now let me ask you a very simple question. Now students, this 4 feet person is now becoming 6 feet now. 6 feet tall. Now just because he became 6 feet doesn't mean that he will become 4 feet now. Tell me yes or no in the chat. Do you students tell me? Tell me students quickly. Right. If this person is becoming 6 feet now, does it mean that he'll become 4 feet? Not really. Because the same DNA for him to be tall is present in every single cell. It's present in every single cell. So this is your entire concept of biotechnology on the board. And if you understand this every single step, isolation, how do we cut it here? Transferring to vector. What is a vector? Transformation. There are multiple methods of transformation. Ballistic method, gene gun, many methods are there. And finally, how the host is reacting with it. This right here is your entire chapter. Right. Students, let me know in the chat quickly. Are you ready to start the entire chapter now? With this basics in mind, all of you have some idea about this chapter in your mind right now? Some idea? Yes, that's all is required. So with this basic introduction, let's start the class now. Because that was the basics. Now students, this entire process of making this DNA here, that is this particular, making this entire DNA and vector and everything, it is called as recombinant DNA technology. Technology used for introducing DNA sequence from one organism to another. Transferring genetic material from one organism into one more organism. What do you call that, students? One organism, genetic material to one more organism. That entire process is called as recombinant DNA technology. So I believe all of you know the basic term now. First term clear to all of you. So if someone asks you what is rDNA technology, it is also called as what, students? rDNA technology. rDNA technology. That is recombinant DNA technology. That is technology used for introducing DNA sequence from one organism to another organism. This entire process is called as rDNA technology or DNA technology. Okay, recombinant DNA technology. Now students, who were the two scientists, right? Who were the two scientists who did the pioneer work? I mean the starting work in this entire industry. The main two scientists were the Stanley and Cohen and Herbert Boyer. These are the two different scientists who did the initial work on recombinant technology. Again students, ready for one more story? Ready for one more story now? Now listen to me very carefully. Now second story. Okay, how does it happen here? These two scientists, their main work was on E. coli and Salmonella typhimurium. Okay, these were the two organisms which these two scientists took. Okay, now what did they exactly do? Listen to me now. They took first of all Salmonella typhimurium. Why? Because this Salmonella bacteria has a restriction enzyme gene. Okay, there's a gene here which is able to produce restriction enzyme, some kind of type of restriction enzyme. So they first took this Salmonella. Listen to me now. They first took the Salmonella. For example, this is your Salmonella plasmid. Now they noticed that this particular not restriction antibiotic. Okay. They noticed that there is a antibiotic resistance gene here. Now this antibiotic resistance genes, first it is the first step. First step is to cut it out. This is the plasmid of Salmonella typhimurium. So first step is to cut it out. So let's cut it. You cut it with the help of restriction enzymes. Our detail we'll talk about later on. But remember, there's something called as restriction enzymes. Restriction enzymes help in cutting. They're like big cut scissors they are. Okay, so you cut it. Now this is your antibiotic resistance gene present from Salmonella typhimurium. Now here is one more plasmid from Salmonella again. Now here in this plasmid we are cutting this part. Again we are cutting this part here. And with the help of DNA ligase, we are adding to the vector plasmid. This is a vector plasmid, we are adding it here. Now we are taking this vector plasmid, right? We are taking this vector plasmid, which is now called as a recombinant DNA. Why? Because it has taken the foreign DNA, recombinant DNA, and we are sending it to E. coli now. So let's quickly summarize what we did here. First, we took a plasmid from your Salmonella typhimurium, isolated the antibiotic resistance gene. Done. Then we took one more different type of organism, that is one more Salmonella, got the plasmid here, that is the vector plasmid, combined both of them using DNA ligase, join them. Then we have the recombinant DNA, and we transfer the recombinant DNA into E. coli. Now once the E. coli is taking it up, E. coli is now becoming what? Recombinant E. coli. That's all. The simple process was used by Haber, that is the two scientists. They noticed that when you do the simple process of transferring, cutting, joining and transferring it, a desirable gene of interest can be easily be transported into a bacteria. Right? Very easy, right? Tell me, is it easy or not? Simple experiment done by your recombinant technology. This entire thing is called as recombinant DNA technology. We are using genetic material from one organism into one more organism, from one to another one. That is called as recombinant DNA technology. Okay, so easy. Just look at it. This entire diagram makes it too easy for you to understand also, right? Now, it may look very easy, students. It may look very easy. All you're doing is cutting, adding it somewhere. But is it actually that easy to do in the lab? Not really. But is it to study? Yes, it is very easy to study. Okay, now we'll undergo something called as steps in rDNA technology or recombinant DNA technology. There are three main steps I told you. First step is what? Identification of DNA with desirable gene. Like how bro was very tall, so Macha went and took the desirable gene, he took that. In the case of E. coli, we took the antibiotic resistance gene, we took that. That is identification of gene. Done. Introduction of identified DNA into the host, that is called as what, students? This is called as transformation. Transformation. That is the second step. Third step is what? Maintenance of introduced DNA into the host and transfer of the DNA into its progenies. That is making sure Macha's offsprings are also very tall. Simple. Okay. There are three main steps in rDNA technology: identification gene of interest, transferring it to the host, and making sure it is multiplying inside the host. Three steps clear to all of you? Three steps clear to all of you, students, in rDNA technology? Now, students, now when I said I have to transfer it to the host, is it really that easy? No. Because there are multiple tools here. Multiple tools. One by one you have to learn the tools. There are tools, something called as restriction enzymes. I'll tell you what is it now. What is ligase? Basically the gum. This is a scissor, this is the gum. What is a vector? What is a host organism? All of these are called as different tools in your rDNA technology. The first one is called as restriction enzymes. First one is called as restriction enzymes. Okay. Restriction enzymes are used to cut the piece of DNA. Okay. Now, students, restriction enzymes. Now, they are called as restriction enzymes or restriction endonucleases because they mainly cleave the nucleic acids, DNA or RNA. Now, there are two types. Okay, listen to me very carefully, students. There are two types. One is called as restriction exonucleases, restriction endonucleases. What is the difference? Listen to me. Very simple it is. First, let's talk about restriction exonucleases. For example, this is a piece of DNA. Let's say this is the piece of DNA. Restriction exonucleases will always cut the DNA at the very ends. Exo means outside. So restriction exonucleases will always cut on the outside. Now, what is restriction endonucleases? Understand. Restriction endonucleases will always cut. Restriction endonucleases will always cut right in the middle. Somewhere. So exonucleases on the outside, exonucleases on the outside, and endonucleases on the inside. Very simple difference. Okay, very simple difference. But students, what if I tell you under restriction endonuclease also there are two types. There are two types here under endonuclease also. Listen to me. For example, the first one. Let's draw it like one large cap I need because it has to be drawn very well. 1 second. Two types here. Okay. First one, you understand what is the first type of restriction endonucleases. For example, let's take a DNA strand. But this DNA strand has a sequence here. Okay. The sequence is very simple, GATTC. The sequence is GATTC. GATTC. Okay. Now, students, here there will be again GATTC. Now, students, this is a particular DNA strand. This is a particular DNA strand here we have. Now, if you pay very close attention, you can read this strand from GAATTC. From this side and this side, it is almost the same. It is the same. If you read from first side or back side, it is going to be same to same. Such a sequence is called as palindrome sequence. It is called as palindrome sequence also. Okay, it is called as a palindrome sequence. First of all, let me tell you, before palindrome sequence, such sequence which can be identified by the restriction enzyme, they are called as recognition sequence. Now, I'll give you a small exercise. Okay, small exercise. How does it cut? Okay. Now, how many of you go to supermarket to buy ice creams or Biscuit is very easy. Okay. So when you go to supermarket to buy biscuits, how do you buy the biscuit? Do you see the first biscuit you take it or no? What do? You go around the entire supermarket. You'll go. Oreo is here. Parle-G is here. Hide and seek is here. Milk Bikes is here. Okay. You keep on walking. One by one you look at every single biscuit. Like a nice look you'll give. Which is more looking nice from the cover. Okay. So you're going and you'll scan. Basically you'll scan the entire biscuit counter. After that, once you find your favorite biscuit, tell me in the chat what is your favorite biscuit also. Tell me in the chat. Okay. Mine is Milk Bikes. Okay. Once you keep on traveling, keep on scanning. Once you find your favorite biscuit, what do? You go and pick it up. Same concept applies here also. What happens here is your restriction enzymes, it will keep on scanning the entire piece of DNA. It will keep on scanning. Like how you're scanning, right? Like you're scanning everywhere. Same thing is happening here also. It is scanning everywhere. Now, once it is finding the recognition sequence, also called as restriction sequence, which is palindrome in nature. Okay, palindromic in nature. It is scanning, and once it's find it desirable, once it finds its recognition sequence, it will come and attach here. So all of you understood the concept? All of you understood? Like how you will scan the entire biscuit counter to find your biscuit, it is also scanning the entire thing, and finally it is finding the recognition sequence. Now understand, once it finds the recognition sequence, how does it cut? That is more important. Okay. Now students, there are two types of cuts here. First type of cut done by your, let's say an enzyme, let's take an enzyme, what is this? 𝐂𝐴R1. Let's take an enzyme, 𝐂𝐴R1. Let's take 𝐂𝐴R1. Okay. Let's take 𝐂𝐴R1. Roman number, right? Let's take 𝐂𝐴R1. Right? 𝐂𝐴R1. Okay. 𝐂𝐴R1 will look for the 6 base pair. Okay. 𝐂𝐴R1 will look. Now this it'll exactly cut here. From here. So students, tell me, is it cutting right in the middle somewhere? No. Once it identifies the sequence, it is cutting somewhere away from the center. Yes or no? Tell me in the chat, all of you. It is cutting away from the center somewhere. Yes. Away from the center. Okay. 6 base pairs, right? Now students, let me draw it for you one more time. What are the DNA sequence? What do we get after this? After cutting, what do we get? Okay, after cutting. Okay. Here what do we have? No. Here we have G. Rest everything is cut. Nothing is there. Then C, T, A, and A. This is the remaining part, because this A, T, C and this G has been completely removed and thrown off. So this is the remaining part. This is the remaining part which is left right now. Now this is done by 𝐂𝐴R1. More I'll tell you about later. Basically find this. Okay. Now this is your remaining part. Now students, here I have always used the same analogy to remember this. I also tell you the same thing now. Okay. Now if you're in a relationship, if there is a breakup, now are you single or you're mingle? Tell me. You're single, no, after a breakup. So same thing happens here. Single. It is single here. Can you see here? It is completely single. Okay. And students, once you are single, you are ready to mingle, you are ready to hang out, somewhere, ready to hang out. So this particular strands, which is very single here, which is very single stranded, which is single here, it is in something called as hanging ends. What do we get here? Hanging ends. Okay, they're called as hanging ends. Okay, they're called as hanging ends, not Pringles. Pringle, it seems. Oh my god. Okay, this is mingle. Okay. Now this is hanging end. Hanging it is. So it's called as hanging end. It can also be called as sticky end because it's right to stick. It is called as sticky end. It is also called as sticky end. So hanging end, sticky end, also called as cohesive ends. Extra term you don't want, remember this much. Okay, also called as sticky ends. Okay, hanging ends. Now this is the one type which happens in the case of restriction endonucleases. Clear all of you? Clear? Cohesive ends also, it can be also called as cohesive ends. Okay. Now second one you understand. Second one. Okay, second one you understand. Small one. Let's take. C, 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 G, G, and G. Okay. So this is the one sequence. So what do you have in the bottom? Here we will have C, 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 right? More. So here we have C, 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 G, G, G. Now here we have an enzyme called a small one. So small one. Okay. Now understand, students, what is happening here. Now unlike here, which is being cut, which is cutting somewhere away from the center, here it is cutting right in the middle. Right in the middle it is cutting. Here. Right in the middle it is cutting. Now once such cut is made, what do we get? C, 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 separate here. G, 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 separate here. C, 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 and G, G, G. Now students, here, now imagine if you are approaching a girl, okay, and she is, instead of making some excuses like no, not interested, she can give you a direct answer. The other name of this direct answer is called as blunt answer. So here we have directly it is getting cut, directly it is getting in the middle. So can I call it as blunt end? Blunt end. It is called as direct answer. Bluntly she'll tell you no. This is also called as your blunt end. Bluntly she'll tell you blunt. Now students, have you heard this famous line? The worst she can tell you is no. Have you heard of it? Have you ever heard in your life that worst thing she can tell you is no in anywhere? Worst thing she can tell you is no. But students, let me tell you, there are worse things. What is the first thing? She can ask you, is like, go flush yourself. Imagine you tell her something and she tells you, go flush yourself. How embarrassing that would be. So remember, why did I tell you that story? Why flush yourself? Because this blunt end is also called as flush end. It is called as flush end. Is also called as flush end. Okay, flush end. Okay, so that is the famous thing. That is two types of restriction endonucleases. One is cutting right in the middle. One is cutting somewhere away from the center. One is getting your hanging ends or sticky ends. Here we are getting blunt end or flush ends. Difficult? Tell me, is this difficult to understand? Tell me, is this chapter even difficult to understand? You understand the entire, right? You understood the entire with the help of Macha and Bro, the entire concept. Now you understood the entire restriction endonucleases, two types. Okay. Restriction endonucleases are enzymes found in certain bacteria that cut the DNA molecule at or near specific nucleotide sequence. So they will find something and they'll cut it there. Now students, do you want to know the story? Do you want to know the story how restriction endonucleases, restriction enzymes came into existence? Do you want to know? Friends, tell me. Do you want to know it? It's very fascinating and it will also give you a better perspective of appreciating bacteria. Want to know? Listen, if you say no also, what option do you have? Huh? What option do you have? Listen to me now. How did the restriction endonuclease or restriction enzyme came into existence? For example, this is a bacterial cell. This is a bacterial cell. Let's take this is a bacterial cell. Okay. Now in this on this bacterial cell, what happened is, let's take, you know who is the organism which will infect even bacteria also? Who knows that? That is called as bacteriophage. Let's say a bacteriophage is coming and it is putting a tent here. It is putting a tent here. Okay. Now this bacteriophage, right? This bacteriophage right here, it will inject its genetic material inside. Inject the genetic material. Now students, this particular genetic material is there, right? Now here, let's take, we have its own genetic material. This bacteria has its own genetic material right here. Now imagine students, let's say Evening or Daisy, all of you, any one of you, if some robber is coming to your house, if a robber or if a thief is coming to your house, do you fight back or do you be like, Anna, come here, Anna, 1 minute, you come, take this gold, take this 1 kg cash. In my bathroom, if you go on the shelf behind the soap box, I've kept more 1,000 rupees. In the kitchen, if you go, my mother has kept more money, take it from there. Do you say that? Do you say, students? If you say that, I don't know what to tell, but do not say that. Okay, do not say that ever. So you never say that, take all the money and go. You don't say that. What do? You're like, you fight back. You fight back with your life. Okay, you fight back, right? Same thing is happening here also. What is happening here? Understand. This particular bacteria is fighting with this. It is fighting with this particular genetic material. How is it fighting? It is producing the restriction enzymes. It is producing restriction enzymes. It is producing restriction enzymes, and this restriction enzymes is going and attacking the chop. Complete chop is happening here. Chop, and it is completely destroyed. Completely destroyed. Gone. Destroyed completely. Now, what are the chances? What are the chances that this restriction enzyme will also attack its own genetic material? There are chances. The thief can be in the house only, no? Your own, some old relative might be coming to steal your own house. Okay. So, restriction enzyme can attack its own genetic material. It can attack. So, there's a solution here. What is the solution? Okay. That is, this particular bacteria is very smart. To its own genetic material, there will be addition of methyl group, or it is called as methylation. It is called as methylation will happen. So with the help of methylation, this restriction endonuclease will not attack its own, it will only attack the genetic material. And right now, we are exploiting this. What are we doing? We are exploiting this restriction enzymes for our own benefit. All of you understood? All of you understood, students? All of you understood? Very simple, very simple. Okay. Restriction endonucleases. Discovery of restriction endonuclease. See, bacteriophage, I told you. This is the bacteriophage, ultimate boss. It is infecting the, see, restriction enzymes. Students, it looks cute here, but it is not. This particular person, restriction enzyme, is going and chopping the entire genetic material here. Okay, looks cute, but it is not cute, but deadly it is. Okay, this is a modification I told you. So when this is coming to attack its own genetic material, modification enzyme is protecting it. It is protecting it. Okay, so cut is the foreign DNA, restriction enzymes. Here we have the modification enzymes. Modifies the bacterial DNA to protect it from against the action of restriction enzymes. Okay. So restriction modification system was discovered in E. coli. Remember that point. Remember that point. All of you will remember. All of you are very smart. Okay. All of you are very smart. Now students, action of restriction enzyme I already told you. Let's take a small one. Small one will cut in right in the middle. I told you. Cut right in the middle. Okay. It will find the recognition sequence. I told you. It will go around like how you go scan the entire biscuit counter, finding your favorite biscuit. It will also scan the entire DNA sequence until it finds its own recognition sequence. And let me tell you, students, let me tell you. This is very important. More than 900 restriction enzymes have been isolated over 230 strains of bacteria. From 230 bacteria, we have 900 restriction enzymes. And the first one was called as HIND2. The first one is called as HIND2, which is 6 base pair 1. The first one was HIND2. Okay, HIND2 was the first one. I think 1963. HIND2. Okay, HIND2 1963, 6 base pair 1. 6 base pair 1 was identified. Okay, that's 6 base pair. Now this is called as palindromes. Read from here and here. Meaning is same. Okay, palindromic sequence done. Blunt and sticky end. All of you understood what is blunt and sticky end. Okay, blunt ends. Right in the middle is called as blunt end. What is sticky end? EcoR1. This is called as sticky end or overhangs. Overhang or hanging ends. Can you see? Hanging ends. Okay. Here's a list of all the different types of enzymes and which type of they might ask. So they might ask you, they might not ask you, but for the safer side I've given to you. And students, this entire notes, all of these notes, see, this takes a lot of time. All of you are aware, right? To make this entire presentation, I probably took around 4 to 5 hours. right? This entire thing will be available on the Telegram channel. Okay. No. Now this is what is given your NCERT. Now here I will tell you one small thing here. Okay, listen to me. This is a small thing which you have to focus here. Action of restriction enzymes. The enzyme cut both DNA strands at the same site. Now what do I mean by DNA site? Both the strands. When you are trying to cut a piece of DNA, that is vector DNA and foreign DNA, you're supposed to always use the same restriction enzyme. You cannot use one for here, one 𝐂𝐴 here and hint to on something else. No. Because when you do the same restriction enzymes, proper hover angle will be produced and you can join them. This line is very important, all of you. The enzyme cuts the both DNA strands, that is vector DNA and foreign DNA. Same restriction enzyme needs to be used. And here the example given is 𝐂𝐴 cuts the DNA sequence base between G and A only. Okay. Now once you cut, sticky ends are produced. They're produced. Okay. Sticky ends is produced. And these sticky ends, okay, like what are they actually cutting here? They're cutting the sugar phosphate backbone of the entire DNA. All of you know the DNA molecule, right? The sugar phosphate is broken down. Okay. Now once you have the sticky ends, they are joined together with the help of DNA ligase. They are joined together. And finally we have the recombinant DNA. Recombinant DNA has the vector DNA and the foreign DNA. That is called as recombinant DNA. See, this entire diagram makes sense now to all of you. Same thing is given in this one also. Can you see? Foreign DNA, vector DNA, students. Now students, again, do you use the same enzyme in both of them? Yes. To cut this foreign DNA and to cut the vector DNA, we use the same restriction enzyme. You cannot use different scissors. Okay. Now, one cutting here. Now, students, do you remember this fancy scissors where you cut, you get designs? If you cut that fancy scissor and normal scissor, you cannot join them later on. We need to use a same type of scissor here. Okay, same type of scissor. Once you make the cuts, join them using DNA ligase given here. Now, once you made the DNA, recombinant DNA, it's called as recombinant DNA. Once the DNA is added to the vector, it is called as recombinant DNA. Recombinant DNA is added to the cell, that is called as transformation. I already told you, transformation. And then it is dividing into multiple cells. Now students, this is a small concept right now. Small concept called as nomenclature, right? For example, all of you have name. Like my name is Baswaraj Rajshekar Shirgumpi. Very long name. I have a first name, middle name and a last name, right? Tell me your name in the chat, students, all of you. What is your full name in the chat quickly? Your full name in the chat. Okay. So all of us have names, right? All of us have good names and names signify something. For example, my name, right? My name means the king who has a power of bull. The king who has a Baswaraj. Raj means king. Baswa means bull. The king who has a power of bull or Baswanna, right? So every single name has a significance. Like you might, let's say Kartik. Kartika is the god. Right? God is signifying that. So every name has a significance. Similarly, right? Here also, every single name also has a meaning here. A plasmid also has a name. Vector also has a name. Okay. Now, see here. For example, the first one. In your restriction enzymes, the first letter signifies the genus name. First letter of genus name. Second one signifies the species name. So first is genus, second is species. Third one is the strain type. That is which strain it has been isolated from. Right? And the fourth one is order of discovery. One quick example I'll give you. Listen to the example. 𝐂𝐴. What is 𝐂? E stands for Escherichia coli. E of Escherichia coli. That is the genus. This is the genus. Coli, the C. coli is the species. That is CO. 𝐂. R is the strain. It is RY strain. So R is being used from there. R. What is the one? That is the order of discovery. It was discovered in the first go itself. So 𝐂𝐴. Understood? That is called as nomenclature. Now, do you have to learn this? Yes, you have to learn this. Questions will come from here for sure. Okay, 𝐂𝐴. Okay. One more example, hint 2. H is Haemophilus, influenza. H from here, genus. I in from the species. D from the strain type, D strain, and the 2 is the order of discovery. Now students, if there is Hind 2, do you think there is Hind 1 also? Do you think that? If this is Hind 2, do you think there is a Hind 1? Yes, it is. Yes, it is. Okay. In fact, the first discovery was done in Hind 1. Isolation was done in Hind 2. Restriction enzymes. Okay. Hind 2. Okay. Now students, what is DNA ligase? All of you are aware DNA ligase is the molecular gum. You use DNA ligase to paste things. Paste it. That is DNA ligase, to paste. Okay. Two different structures here. You add the glue, you fix it. That is DNA ligase. Okay. Now students, do you realize in just 1 hour, your 40% of the chapter is actually over. How many of you realize that? How many of you realize that, students? In just 1 hour, 40% of the chapter is over. All of you drink some water now for the 40% completion. Okay. Students, I want everyone to quickly smash the like button. Let me check the likes today, right now. Let me check the likes. Students, 125. What is this? I will get scoldings later on. Quickly, all of you. Fire emoji in the chat, all of you. If you're able to follow till now, if you have any doubts till now, quickly tell me in the chat. Because this is a chapter which can either make you understand concept or make you forget everything. Okay. Do you realize 1 hour just went away? I didn't realize how 1 hour went away. I don't know why. I just saw the time some time back and I'm looking here and I'm looking again back. 1 hour just went away. It was just like so fast. Yeah, application will also happen today only. Don't worry, Ravindrani. Right? 1 hour just went away like a magic. That's the best part about learning, right? When you learn in such a engrossed environment, you don't even understand how 1 hour goes away. It just magically goes away. Right? All of you drink some water. So with that, can we start the second part of this chapter now? That is gel electrophoresis. Can we start? Can we start now? Can we start? Yes. Okay. Now students, once we have our piece of DNA after cutting, right? After cutting the segment of DNA, we need to find our gene of interest from that DNA, right? Like once we get a long piece of DNA, we can't keep on replicating the entire DNA, not possible. We need that specific piece of DNA, specific gene of interest that needs to be replicated eventually. So to separate the DNA strands, there's a process called as gel electrophoresis. Gel means in gel media. Electrophoresis, electro means electricity and forces means being carried. So we are using electricity in a gel media to separate DNA strands. And the separation of DNA strands is based on the weight of DNA strands. That because there are certain segment of DNA which is heavier, certain segment which is lighter. Lighter will go further, smaller segment will go further, heavier segment will be on the back. Now students, do you want me to teach you the briefly electrophoresis or do you want in detail by becoming a scientist? Tell me in the chat now. Tell me in the chat quickly. How do you want to learn it? Do you want to learn it like textbook style? Textbook, they would have hardly given you 2-3 lines, mug up that lines and you're good to go, or do you want to actually learn it? Like how it is done in lab? Because I will be teaching you from a personal experience, not from the textbook. Not from a textbook. I will be teaching you, I have made this from my own personal experience, like how I used to do it on a daily basis. Right? How I used to do it on a daily basis in the lab a couple of years back. Yes, how do you want to learn it? Students, you know, right? All these are theoretical, rhetorical questions. Even if you say no also, what will I do? Tell me, students. Even if you say no, you're like, no, sir, textbook is enough. What will I do, students? Tell me. All of you know me. All of you know me very well. I will teach you detailed only anyway. Okay? All of you know me very well. Now, students. First of all, you need to understand the charge on the DNA. That is very important to understand. Can anyone tell me in the chat, what is DNA? Is it positive or negative? Tell me, students. Is it positive or negative? First question to all of you. I want everyone to be active now. Only if you're active, then it makes sense. I want everyone to answer. Okay, tell me. DNA is negatively charged. Okay, DNA is negatively charged because of phosphate group. All of you are aware, because of phosphate group, DNA is negatively charged. Negatively charged. Okay. Now first step is to make the gel. Before we add it into the entire media, you need to make the media first of all, make the gel. This right here is the conical flask. It is called as a conical flask. In this conical flask, we need to add the agarose gel, agar. Now students, I want everyone to answer this question. Agar is obtained from which algae? Let me give a small clue. It is part of your red algae. Answer in the chat. Red algae. Okay. Tell me the proper examples. Plant kingdom. Okay. So this is your agar obtained from the algae. Okay. Now students, can you see all of this? This is called as this is entire thing is the slot where we add the piece of DNA and the DNA moves here. DNA moves here. Okay. DNA is moving with the help of a sieving effect. With the help of a sieving effect here. Okay. Sieving effect. Now students, I told you the principle already. This is negative cathode. This is the anode. So DNA being negatively charged, it will move towards the anode. Yes or no? So large DNA will be at the back, small DNA will be at the front. That is the movement of DNA. Okay. Now, first of all, you need to melt the agar. No one tells you this. NCERT doesn't tell you this. It is coming from personal experience. Unless you completely melt the agar completely by shaking it vigorously, keeping it in microwave, it is useless. Let me tell you. Once you take it out from the media, you're supposed to pour it after a few minutes. You cannot pour it directly because it will create a fog. You cannot add very late also because it will again become solidified. So you have to find the right time to add it. Okay, that is your thing. Okay. Now, to this you need to add a coloring agent basically to observe the DNA called as ethidium bromide, which provides a bright orange color. Ethidium bromide. Okay, ethidium bromide, that is ETBr, is added. It helps to visualize DNA because DNA is naked. We cannot see the DNA with naked eyes. Ethidium bromide. Okay, ethidium bromide is added. Okay. Now students, this is called as the cast or a tray. Okay. In this cast or tray, we add the entire DNA is added here. Entire DNA, not entire agarose is added, not DNA, agarose is added. Now students, do you see this entire part here? This is called as a comb. Not your Rajnikant comb, not this comb. This comb is different. This comb is used to keep it here and to make the small wells, to make something called as small wells. Okay, so add that. You add the comb here. Now you let it solidify. Once solidification is done, you remove the comb out. Once you remove the comb out, do you see this? These are called as wells. They are called as wells. Okay. Now once that is done, you add the entire cathode and anode. We also add a buffer here. Running buffer is also added. Now students, loading the DNA sample. Okay. This usually generally no one tells us. I don't know why, but this is a very important step. You cannot observe the movement of DNA on the tray itself if you don't add something called as a loading dye. If you're my student, you should be able to tell this. There's something called as a loading dye. We add something called as a DNA loading dye, which is generally blue in color. It's called as DNA loading dye. So we mix DNA loading dye plus DNA. Once the mixture is made, that mixture is added here. You cannot add the direct DNA. Not possible. Okay. Not possible to directly add it. Okay. So you add the DNA plus the loading dye because we need to see the movement of DNA, right? That is only possible because of loading dye. Now you might ask me, sir, what about ethidium bromide? Student, let me tell you, the use of ethidium bromide is towards the end, not here. Okay, so you add the loading dye. Okay, now once you add the loading dye, you set up the apparatus to the electricity and you make it run. Okay, you make it run. Now students, let me show you a small video. All of you can see, this is how the case is put. This is the entire equipment. All of you can see this entire equipment. Here we add this cathode, anode, and there is a buffer here. The entire buffer is also added. Okay. Once you start the electricity is running. It's running. Okay. Now can you see? The DNA is moving here. DNA is moving. Can you see the movement of DNA? The blue color. This blue color is because of loading dye. Because of loading dye, not because of ethidium bromide. Not because of ethidium bromide. This is a loading dye. So we can see the movement of DNA. DNA is slowly moving out based on the size. Okay, students, that was the entire DNA. That is basically adding the DNA. Okay, now once you add the DNA into the well and everything is done, right? Now finally is visualization. Visualization is done with the help of ethidium bromide. Final step. Now students, you cannot see the entire DNA on its own. Here what you need to add is a trans illuminator. We need to add something called as UV rays. Okay, UV trans illuminator. Can you see? With the help of UV trans illuminator, we can visualize the DNA. We can visualize the DNA under the UV trans illuminator. So only under UV trans illuminator, because of ethidium bromide, it is radioactive in nature. It is radioactive. It is carcinogen. Radioactive in nature. We can visualize. Now you might ask me, sir, yes, separation is done, but how do I know in that entire separation which one is my desired DNA? You might ask, right? See, after the entire movement, after looking at the trans illuminator, UV trans illuminator, this is how I get. How do I know that this one right here is my gene of interest? Small thing. Let me tell you. Because there is something called as a DNA ladder. With the help of DNA ladder, you can match. For example, this is the entire slab which you get. Here is your gene of interest, and all of us, when we cut and make the segments, we know the size of a gene of interest. You simply correlate. So here, let's say this is around 1500. So let's around 1400 is the gene of interest. You mark it out. Simple as that. From here to here you compare. Like how you take one scale, no? Like let's say this is my gene of interest. You know your gene of interest, let's say around is 7, 8, 10 centimeters. Your gene of interest is around 10 centimeters. You take a scale, you measure 10 centimeters. You got a gene of interest. This one, let's say, is around 20 centimeter, 30 centimeter, 5 centimeter, 6 centimeter. You can't take any of that. You have to only take your gene of interest, which is around 10 centimeters. So you take a scale next to it, measure, compare both of them, and then take this one. All of you understood how to take your desirable gene of interest? All of you understood how to take your desirable gene of interest? That is all. Now, once you know this is your gene of interest, once you know that right there is a gene of interest, next step is called as elution. It is called elution. You add a suction cup and you remove the DNA out. You remove the DNA out completely. Okay, remove the DNA out. You take it out, add it into a Eppendorf tube. This right here is the Eppendorf tube. Once you add it into Eppendorf tube, you give heat shock. Why? To melt the gel entirely. Obviously, we need to melt the gel. Okay. Now, after melting, we need to precipitate DNA out. How do we do that? For that, we have a simple method called as chilled ethanol. Once you add the chilled ethanol, entire DNA is precipitating out. It is completely precipitating out. Can you see here? And for example, if our gene of interest was insulin, you finally get the insulin with the help of your gel electrophoresis. So all the way from DNA is being negatively charged, DNA making the agarose gel with the help of your media, setting up this entire thing. What is the principle? DNA being negatively charged, it will move towards the anode, positive charge. Small will go further, medium, large will be at the back, melting it, adding the comb, adding the media, adding the ethidium bromide, adding the loading dye, adding the ethidium bromide, loading it, running it later on, electricity. Then it is moving based on the size. Then we are using UV trans illuminator to visualize under the microscope, under the UV transluminator. Then we compare and get the gene of interest, and finally we remove it out and we keep it in a Eppendorf tube. In this tube we add it in this tube, and once you have the gel, you melt it completely, then add chilled ethanol, and finally DNA is precipitated out. DNA is finally precipitated out. So that was the entire steps in the gel electrophoresis. Yes. Okay. Done, all of you? See, Gopika ma'am is live on the chat. I think Gopika ma'am is also Gopika ma'am has given me target also. So students, 200 likes before the session ends. Before I go also, 200 likes should be there. Okay. Now students, with that, entire gel electrophoresis is also complete. Okay, it is completed. Okay. Now students, next thing is insertion of gene of interest into the vector. Like how do we take our gene of interest after isolation? After isolation from gel electrophoresis, we need to take the gene of interest and add it to a vector. How do we do that? Okay. Now, first of all, you need to understand what is a vector. Vector is basically a taxi. You cannot take, I repeat again, you cannot take our gene of interest and directly add it into a host. From here to here, you can't add it. Before you add the gene of interest into the host, it has to go through a taxi. You cannot jump and go to your friend's house. No. What do? You have to take a vehicle. If the friend is around 10 kilometers far, you will not walk and go also. You will not jump and go also. Not possible. You will take a cab, you will take an auto, you will take a bike, car, something. You will use a vehicle to travel. The vehicle here is a vector. Vector is basically a vehicle which is helping you to transfer the DNA. Okay, helping you to transfer the DNA. Okay, so DNA sequence into which gene of interest can be inserted is called as vector DNA. It acts as a carrier that carries the gene of interest into the host where it is multiplying. Okay, so we have a gene of interest after gel electrophoresis and everything. After isolation, we are adding it into a vector. Here. After addition, this vector is added to where? This vector is added to the host. We can't directly add it to the host. Not possible. Add it to the vector. Vector will go into the host. Okay. Now, what two type of vectors? Right. There are roles of vectors. First, all of you know carrying gene of interest. That is the first role. Second role is producing copies. That is also a role of vector only. Once a vector goes inside, once the vector plasmid, for an example, once it is entering into the host, it is also responsible for making multiple copies of it. Okay. Now students, there are different type of vectors. Now students, all of you should understand the difference between vector and a plasmid. Vector is a broad term, vehicle term. Like under vehicles you have car, bike, scooty and everything. No. Similarly, we have multiple types of vector. Plasmid is a type of cloning vector. Okay, it will help in making multiple copies. Once this particular plasmid is inside a host, it will not only transfer genetic material, but it will also help in what? It will also help in making multiple copies inside. Why? Because this plasmid right here has something called as oocyte. I will tell you later on what is the oocyte and everything. Okay, so plasmid is a type of cloning vector. So please remember that. Okay, plasmid. Okay, what is plasmid, students? Extra chromosomal DNA and external DNA present in cells other than genetic material. For example, this is a bacterial cell. In this bacterial cell, we have the normal DNA, bacterial DNA. Apart from bacterial DNA, inside the bacteria, there is an extra chromosomal, self-replicating DNA. That is plasmid, right? That is a plasmid, right? Now, circular DNA, which circular in shape, present in bacteria and some yeast. That is plasmid. Now, what is phages? Phage is also a type of cloning vector known as bacteriophage. So we can also use bacteriophage to infect bacteria and also transfer genetic material. Okay. Then what is BAC in yak? Understand. BAC is nothing but bacterial artificial chromosome. Yak is yeast artificial chromosome. Both are type of cloning vector. BAC is obtained from bacteria. Yak is different from the yeast. That is all you need to know. Remember students, both are genetically engineered. They have been created by us, not naturally present. They are not naturally present, they are genetically engineered. Okay. They are used in Human Genome Project, HGP. Used in HGP. Now students, next question to all of you. Can any random DNA sequence be cloning vector? Can I take any random? Can I take any random? Can I take any random cloning vector and call it cloning vector? Or are there any properties of cloning vector? There are properties. Okay, listen to me. What are the properties? It should have ability to self-replicate. That is why I told you. A vector should be able to replicate inside the host. Okay. It should have repressor of primer, also called as repressor of plasmid. Should be able to produce replicating the plasmid. Okay. Should have selectable marker. I'll come back to this. What is selectable marker? I'll teach you today only. What is blue and white screening? What is selectable marker? I'll teach you today. Okay. It should have a cloning site. Now, what is a cloning site, students? Cloning sites are the sites where the restriction enzymes will come and cut also. Okay. How many of you know about this human celebrity? How many of you know the celebrity here, all of you? How many of you know the celebrity? How many of you know the celebrity? Heard about this PBR 322? Anyone of you know? PBR 322, a famous celebrity here. Let me tell you students, on this celebrity, right, there are many questions. Many questions on this celebrity. Okay, many questions will come on this celebrity, so we need to understand the celebrity very well. Okay, very well, you need to decode the celebrity. Every single point about the celebrity we should know. Okay, so can we do that now? This celebrity is called as PBR 322. Okay, PBR 322. Now, first of all, what is the meaning of PBR 322? P stands for plasmid, B stands for Bolivar, discovery, R stands for Rodriguez. 3T2 is a discovery, right? That is the discovery. That is the discovery thing. So PBR 322, right? Now there are few points which you need to know about the celebrity. Few points. So listen to these few points, because now I'll be teaching you something called a selectable marker. That is going to be very, very important. Many questions will come from there. Can we start, all of you? Can we start? Yes. Can we start, students? Can we start? Characters of an ideal vector. Looks so nice now. Looks so nice. So let me tell you, this diagram is very easy diagram given here on the board. It actually looks very difficult. It is not so easy to see. As in, like, not easy to represent. It is much more complex than this. PBR 322, the right way to draw. I remember still. Okay. Now students, remember what you need to understand here first of all. This is a plasmid. Plasmid is a cloning vector. Every single cloning vector, I told you, should be able to replicate. Now students, if a particular structure needs to replicate, it needs to have something called as ORI site. Origin of replication. Here. Can you see? ORI site. It is called as origin of replication. This ORI site will help in replication, initiation replication, and also something called as copy number. Now you might ask me, sir, what is a copy number? Okay, listen to me. Every single plasmid has a capacity to how much it can produce copies. If you want a lot of copies, you should go for a better plasmid. If you want less copies, you should go for one more plasmid. So ORI site also tells you the copy number. Apart from origin replication. Apart from this, students, can you see here? There is something called as ampicillin resistance gene and also tetracycline resistance genes. These two are different types of restriction sites here, and both of these can be used for selectable markers. What is selectable markers? I'll tell you. Selectable marker later on. Now here, can you see different types of slots where restriction enzymes can join? These are different cloning sites. Okay. See here. ORI or origin of replication is a sequence that initiates DNA replication. Done. An ideal vector should have ori site. It should have ori site. Without ori site, it cannot replicate. Okay. This property allows the vector to multiply within the host. Okay. ROP is called as repressor of protein or repressor of plasmid also in many cases. Okay. It's in replication. It has two different types of selectable markers, that is antibiotic resistant genes. Antibiotic resistant genes are there. Okay. We have ampicillin resistance gene and tetracycline resistance genes, both are present here. Two antibiotic resistant genes are present here, I told you. Two are present. Okay. Now students, I'll tell you a small thing here which you have to remember at any cost. You have to remember at any cost is which different types of sites are present in which particular region. In ampicillin we have PUV1 and PST1. Here in ROP we have PUV2. In tetracycline we have BAMH1 and SAL1. You have to remember this at any cost. There is no other go. Now if you want a trick here, let me know in the comment section. I will make a trick for this. If you want, that is. I'll make a trick for this. I'll make a short for it. I'll make a short. Don't worry. Okay. But if you know, very good. Because you have to remember this at any cost. You can use any trick in the world, anything in the world, draw it and learn it. You have to remember it. Okay. Now. Students, all of these sites are the sites where your gene of interest can be added. We can add a gene of interest here. We can add a gene of interest here. Now students, tell me, what if I add a gene of interest here? What will happen? A logical question to all of you. What will happen if I add the gene of interest right here? What do you think will happen, students? Will it replicate anymore? Will this particular plasmid will be able to replicate anymore? Obviously not. It won't be able to replicate. So be careful of where do you add the entire gene of interest. Okay. So it will have restriction enzyme sites or recognition sites where REN cuts. Okay. So we can cut here. REN can cut here. And once the cut is made, here, gene of interest can be added here. It can be added here. Okay. Using DNA ligase. So, okay. That is all, students. That is all is the celebrity. For celebrity, you need to know, students. For this celebrity, you need to know the name, why the name is there. But why, sir? Generally understand, if you cut this region only, it is losing the sequence of replication. If it loses the sequence of replication, it cannot replicate anymore. Okay. Now there is let me tell you, students, there is an alternative to this also. There is a question in NEET 2024, if you remember, there was a question which many students could not answer because the concept was not clear. There are two ways a plasmid will replicate. If a plasmid has to replicate, it has to have ori site. That is the given rule. The given rule is that if a plasmid has to replicate, it needs to have ori site. It needs to have ori site. That is the first thing. Okay. If it does not have ori site, it cannot replicate. Now, students, what if this plasmid is integrating into the host? This plasmid is integrating the host. The host has the ori site. The host has the ori site. Can that time the plasmid, can this gene of interest replicate? Tell me, all of you. See, generally, if I am cutting this part, I am adding gene of interest. Now this plasmid is able to replicate inside the host because of the ori site. And when it is replicating, gene of interest is also replicating. It is also replicating. It is able to replicate. Now, what if there is no ori site at all here? There is no ori site. There is no ori site at all here. But this particular gene of interest is already integrated into the host. Gene of interest is already integrated into the host. Can that time it multiply using the host ori site? Yes, it can. That was the question asked in your NEET 2024. That was the question asked in your NEET 2024. Let me show you the question. Let me show you the question, students. See here, this was the question. See here. What is the fate of piece of DNA carrying only gene of interest? So we have a DNA piece. It only has gene of interest, only has gene of interest, which is transferred into alien organism. So we are taking the gene of interest and we are adding it into the host. Adding into the host. Okay, that was the question. Gene of interest, addition into the host. Now options given to you was the piece of DNA would be able to multiply itself independently in the progeny cells of the organism. Can it multiply independently? No, it cannot. No, it cannot. Why? Because the oocyte is missing. Oocyte is missing. It cannot replicate. Second option C. It may get integrated. See, option number B is very smart. Now they're telling you it may get integrated. Our gene of interest may get integrated in the host. Integration has happened now. Okay. Into the integrated into the genome of the recipient. It can get integrated. It can get integrated. Now, it may multiply and be inherited along the host DNA. Now, if it has to inherit into the host DNA, it means integration has already happened. So it can multiply. The gene of interest can multiply only when it is directly integrated in the host. Otherwise, independently it cannot multiply. That is why this option is wrong. It shows ability to replicate. No, it cannot replicate on its own. It cannot replicate on its own because the oocyte is missing. It can replicate if it gets integrated in the host. Okay. The alien piece of DNA is not an integral part of chromosome. No. Okay. So this was a very good question given to you in NEET 2024. Only 50% were able to answer. I know, understand why, because this concept is missing from NCERT technically. Okay, like how vector DNA actually performs. But I taught you this last year. If you remember last year class, I taught you this last year also. Like how without oocyte, how gene of interest can multiply. It can multiply. Okay, beautiful it was. Beautiful concept. Now students, wait. Next thing is students, insertion of rDNA into bacteria. Okay. Now it is already methylated. Generally, it is already methylated. It is already ready. When you make gene of interest, right, you don't randomly send it into war directly. In such cases, it is already methylated. We take precautions to make this entire triplicates, quadruplates, and we test one by one. So to get that result, desirable result. Okay. Now students, next thing is called as insertion of rDNA into the bacteria. Hello sweetie, hello all of you. Right. Next step is called as insertion of rDNA into the bacteria. Now students, again, here we have our gene of interest. Let's say we have rDNA here. Recombinant DNA is ready here. Recombinant DNA is ready. It has this rDNA. Let's say this is the plasmid. This plasmid has our gene of interest here. Gene of interest. So we have inserted this gene of interest into the plasmid. Now students, I am supposed to send this rDNA. Plasmid is ready already. Vector. This is also or vector. Okay. I am supposed to send this plasmid or vector into the bacteria. Now the question arises, students. Will the bacteria keep quiet and take the plasmid, or will it fight back? Tell me in the chat. Tell me in the chat. Can it directly take the bacteria? Can it take like Papa bacteria? Will it take it directly, or will it fight against it? Tell me in the chat. Tell me. Will it take or will it fight? It will fight, obviously. Right? It will fight. It can't randomly take the bacterial DNA. Not possible. Not possible. Okay. So for that, the main reason is the entire surface area, entire surface. If you look at the membrane, of bacteria, it has three protections. It has a glycocalyx, it has cell wall, and it also has the plasma membrane. So we need to bypass all the layers and send it inside. Now, how do we send it inside? Okay. First of all, we need to make sure the polarity is same. Right? Bacterial cells have hydrophobic membrane. DNA is hydrophilic. See, it is fighting against each other. The charge of the DNA and the membrane are negatively charged. They repel each other. Negatively charged DNA, negatively charged, they will repel each other. Okay, the rDNA cannot pass the bacterial directly, not possible. So what do we do? Now the simple solution is add a divalent cation here. Okay, simple solution is once you add the divalent cation, treating the positive divalent cations like calcium, right, changes the charge on the membrane and DNA making it neutral. So it changes the charge here and making it much more neutral in nature. Now once it becomes much more neutral, the DNA, your particular rDNA, recombinant DNA, students. Why am I calling it recombinant DNA? Because it has the gene of interest. Gene of interest. Now rDNA can enter inside. Okay, rDNA can enter inside. Now students, cells that are capable of taking up the DNA are cells which can take up the DNA. Such cells or such bacteria is called as competent cells. Competent cells. It is generally, bacteria will always fight back. Once you add the divalents, a bacteria will become much more softer in nature and it can take up the entire rDNA. So such cells which can take up the DNA, such a cell is called as competent DNA. It is called as competent cells or competent entire bacteria. Okay. Now, next step is called as transformation, students. Transformation. Does anyone know what is transformation? Remember? Sir, if only the foreign DNA is introduced inside the cell, can it go itself and integrate with the host chromosome? Is it possible? Yes, that is possible. That is what we're trying to do. That is what we're trying to do, right? We're taking a gene of interest or we are taking a plasmid. Usually plasmid is used because it is easier to do it, right? Easier for it to enter inside the host and everything, right? Some cases, yes, gene of interest can integrate directly. Like for example, in the case of HIV, students, you have studied HIV, right? HIV you have studied. In the case of HIV virus, when the viral genetic material is inserted inside a host, does it need an extra plasmid there for entry into the host, students? Remember, try to remind my fresh memory. Try to remind me. In the case of HIV, when the foreign DNA is added into the host, does it need an extra vector to add into the host or does it get integrated eventually? Tell me. Because of other, it can happen, right? Yes or no? Reverse transcriptase, RNA to DNA can incorporate, right? So it doesn't need anything plasmid there, doesn't need any plasmid. So there are multiple cases where it can happen, but we use a plasmid, we need a vector because two minutes I told you. Vector will always help in cloning. See, transfer is there. A vector will help in transfer genetic material. But also students, a vector will help in better copy number. It will help in making multiple copies on the inside. We don't have to rely on the host. We do not have to rely on the host to make the copies. Alright. So in life also don't rely on someone else. In exam also, make your own copies. Right? Make your own copies. That is what plasmid is doing. So we generally use a plasmid, okay, or a vector. Now students, next thing is called as transformation. Now what exactly we are doing transformation? What exactly are we doing in transformation? Right. In transformation, now our plasmid is ready. Our plasmid is ready. We are taking the plasmid or the rDNA, recombinant DNA, or vector. We are taking it and adding it into the host. You are adding into the host. Take it, add into the host. Simple as that. Simple as that. That is called as transformation. Okay. Exogenous naked DNA into the desired whole host. Okay. Now there are multiple methods here. Heat shock method, micro injection, ballistics, disarmed pathogen method. Very simple. First is heat shock method. Okay. Heat shock method. Now first we take rDNA with gene of interest, competent bacterial cells. Now students, all of you know what is competent bacterial cells? Okay, all of you know? Yes, it is enough, don't worry. Right? Transformment and I'll tell you. Okay. Now students, this is the competent bacterial cell. All of you know what is competent bacterial cell? Competent bacterial cell is a cell which can take up the DNA. Okay. Now generally when do you use the word recombinant and when do you use the transformant? Listen to me, the usage is different. For example, this is a gene of interest. Okay, this gene of interest, this is gene of interest. It is added to a plasmid. It is added to a plasmid. Now here I will use the word what? I will use the word recombinant here. Recombinant. I will use the word recombinant. Why? Because transformation has not happened yet. Transformation. If I take this particular plasmid or vector and add it into the bacterial cell, that is called a transformation. Now if this bacterial cell is taking up the gene of interest, that cell becomes recombinant? No. That cell becomes, tell me students, all of you in the chat, if this cell takes up the DNA of transformation, that becomes what? Transformant. That becomes transformant. Okay, this is recombinant after adding the entire bacteria. Okay, so simple usage. Okay, now combinant cells, we are adding the piece of our DNA. Now students, we have to shock the bacterial cell now. We have to shock the bacterial cell and do not give it time to react at all. Like completely shock it. How do you completely shock it, students? How do you completely shock it? You take this entire bacterial cell and keep it inside the ice. Then keep it in that water bath, extreme hot condition. Again, keep it in ice. So ice, hot water, ice. Bacteria does not have any time to react to the gene of interest. It will simply take it inside. You do this in the house also. When your parents are asking your marks, what do? When the parents are asking your marks, you'll be like, something you will tell, that story, this story, this story, that story, you'll try to confuse your parents. All of you agree? Do you remember? You try to confuse them. Okay, same thing we are doing here also. We are confusing the bacteria by giving it a shock. Okay, so keeping it hot water, so cold, ice cold, hot water, ice cold. So when it gets completely confused, it is taking up the bacteria. Okay, basically taking up the gene of interest. Okay, no. Transformed, see, now I'm using the word transformed or transformant. Okay, transformed bacterial cells are now plated and then incubated room temperature for the next steps. Okay, rDNA next is called as microinjection. It is microinjection. It sounds exactly like microinjection. What exactly is happening in microinjection? Let me show you a small video. You'll get better clarity. In microinjection, this is the bacterial cell. We are taking an injection. We are piercing the bacterial cell with a needle. Inside the needle there is a DNA, rDNA, and you directly transfer it inside. Nice, no? Nice, no, students? So this is called as microinjection. We are directly inserting inside the host. That is called as microinjection. rDNA directly injected into the nucleus of the cell. Used for animal cells. It's for animal cell. This is microinjection. Okay. Next thing is called as ballistics. Ballistics or gene gun. Okay. Basically, you take a gun which has the gene of interest. Take a leaf, take the gun, keep it there, shoot. With that same energy, same pressure and same velocity, a gene is transferred into the host. Simple, gene gun. Okay, it's called as gene gun. Next thing is called as ballistics. Understand what is ballistics? Now, for example, this is the chamber. This is the entire chamber. This is the ballistics here. This is a ballistic chamber. Okay. Now here, what do? Is we pass here high velocity particles. Okay. Here we have our, let's say our gene of interest is here. Okay. Gene of interest is here. Okay. Now this high velocity particles are coated with tungsten or gold. Okay, tungsten or gold. Okay. Now once this pass through here directly, when it is coming from the high velocity, right? High velocity is coming and it is colliding here. Now what will happen? Entire DNA here, rDNA present here, with the velocity, with the same energy it is coming, velocity is coming, it's smashing here. Entire DNA is being injected into the cell. It's called as ballistics method. We use inert metal like tungsten gold here. Inert metal like tungsten gold for the entire ballistic process. Very simple, students, very simple. Next thing is called as tumor inducing. Like we use gun, we use injection, we are fooling the bacteria. What else? We are also using animal retroviruses, and also we are using plant bacteria also for advantage. We are not leaving anyone. We are not leaving anyone, students. We have to take advantage of every one of them. Now what are we doing here? Now we are taking advantage of a poor bacteria, not so poor for the plants, that is dicot plants, but this bacteria called as Agrobacterium tumefaciens. This bacteria has a simple life. This bacteria has a very simple life, students. This bacteria, Agrobacterium tumefaciens, okay, Agrobacterium tumefaciens has something called as TI plasmid. It has something called as TI plasmid. Okay, it has something called as TI plasmid, also called as transfer plasmid. Has something called as tumor inducing, not tumor inducing. What am I writing? TI plasmid called as tumor inducing. Tumor inducing plasmid. Okay, it has something called as tumor inducing plasmid. This is the entire plasmid present in Agrobacterium tumefaciens. It is a bacteria. Okay, TI plasmid. Now this TI plasmid has something called as tDNA, called as transfer DNA. This is a transfer DNA. Transfer DNA. Okay. Now students, what happens here? Understand. Okay. Let me understand. Okay. Now this particular organism, Agrobacterium tumefaciens, it will infect the dicot plants. Can you see here? This is the typical bacteria. When it is affecting the plant, it's going to plant cell. Once it goes into a plant cell, it is causing the plants to rapidly produce growth factors, oxygen cytokinins, and plants are undergoing a disease called as crown gall disease. Can you see here? This entire swollen part, it is called as crown gall disease. So what do? We take advantage of this, right? Anyway, this particular TI plasmid and this bacteria is able to infect this plant very well. So what do? So what do, students, here? We are very smart. What do we do here is, in this TI plasmid, right, this entire TI plasmid, in this tDNA region, we chop some part. We chop some part, and we add our gene of interest here. In this tDNA region, there's something called various genes and everything. We don't have to delete everything. Some amount is deleted. Some base pairs are deleted. After you delete the base pairs, you add our gene of interest into this tDNA region, and rest of the work is done by the bacteria itself. Nicely, just do the small changes in the bacteria, in the plasmid. After, you don't have to do anything, because this particular bacteria will infect. The plant on its own. It will cause a disease. When it is causing the disease, right, our desired product is also being formed. It is being formed. Simple as that. Okay, that is your tumor inducing plasmid, TI plasmid. So what we do is, TI plasmid, we cut this small tDNA part, we add our gene of interest there. When this is typically infecting the plant, when it is multiplying inside the plant, our gene of interest is also multiplying. So we get a desired product. That's all. We can use this as a vector now. Simple. Okay. Now what are transformers? I told you already. Host with rDNA and transformant. They're called transformant. Oh, this question solves your question also. This is the host. If the host has the rDNA, it is called as a transformant. It's called transformant. Now next question arises is called as selection of transformant. Now how do you select your transformant, students? How do you select a transformant? Okay. Now I want everyone to focus for the next 10 minutes, ultimate attention. Can we start? Next 10 minutes, ultimate attention now. Next 10 minutes, ultimate attention. Can we go? Students? Next 10 minutes, ultimate attention, because I'll be teaching you how selectable markers work. I'll be teaching you how the entire blue and white screening happens. In next 10 minutes or 15 minutes, let's say 15 minutes. Next 15 minutes, I will teach you this entire concept like there's no tomorrow, and you will never forget it. You will never forget it in your entire life. Can we start? Students? Can we start? Drink some water, all of you. Drink some water. All of you, if you're new to the channel, quickly smash the like button. All of you, if you're new to the channel, quickly smash the like button. Right. Let's go. Let's go now. Can we start? Yes. Now, students, if you remember, this is PBR 322, right? 322. Our celebrity is back. Celebrity is back. Now, I told you this PBR 322 has two restriction enzyme sites. Restriction enzyme, right? That is, one is called as ampicillin resistance, and other one is called as tetracycline resistance. Okay. Now, answer to my question. Answer to my question. If I take this particular PBR 322 and I dip it in ampicillin, will it die or will it survive? Tell me. First of all, tell me that much. If I take this PBR 322, if I dip it in if I dip it in ampicillin, will it survive or not? It will survive. Why? Because of ampicillin resistance gene. Now, if I take this and dip it in tetracycline, will it survive again? It will survive again. Why? Because it has tetracycline resistance genes are present here. It is present here. It can survive. So it can, this right now, this can survive in ampicillin and tetracycline media. It can survive. It can survive. Okay. Now, students, what if I have my gene of interest here? I have my gene of interest. This is my gene of interest. Okay. After electrophoresis, I have gotten my gene of interest. I will take this gene of interest and I'm supposed to add it somewhere here. Now, for example, the restriction enzyme I am using is BAMH1. Tell me, where will it cut now, students? Tell me. Where will BAMH1 cut? Will it cut here? Will it cut here? Or will it cut here? Or will it cut here? If my restriction enzyme is BAMH1, it will completely scan it. It will completely scan it. Scan, and it will find a sequence here. It will find a sequence here and it will cut this part. It will find a sequence here. It will cut this part. Yes, it will cut here. Yes, it will cut here. It will cut in tetracycline part here. It will cut here. Now, once this region is cut here, I am adding my gene of interest here. I am adding my, what I am adding here, students? All of you tell me in the chat. I am adding my gene of interest in BAMH1 part. I am adding here. Okay, I am adding it here. Now, students, understand. As soon as I am adding my gene of interest here, right? This entire region which had tetracycline resistance is also lost. It is also lost. Now, what do we have here? Let's take, this is my PBR 322 right here. In this PBR 322, here this side I have ampicillin resistance, but this side I have now tetracycline susceptibility or sensitivity. It is now sensitive to tetracycline. Very sensitive to it. Very sensitive to tetracycline. This one. Now, students, now this is my gene of interest here. Now, this is my plasmid. PBR 322. This is my plasmid right here. Plasmid. Now, can I call it now rDNA also? Recombinant DNA, rDNA also. Can I call it recombinant? I can call it recombinant. Whatever I call it. I can call it recombinant. I can call it my recombinant also. Okay. Right. I can call it recombinant. This is my recombinant now. Now, students, I am taking this plasmid and this is my plasmid or vector or my vector, and I am adding it into a host now. Let's say I have a host here. I am adding it into a host. The process is called as what, students? Tell me in the chat. What do you call the process? The process is called as transformation. With the help of transformation, I am sending this into this. You can use gene gun, we can use a ballistic method, micro injection, right? Heat shock method, tumor inducing, any method we can use and take this rDNA, this recombinant DNA and entire plasmid, add it into the host. Add it into the host. Transformation done. Transformation done. Okay. Now, once transformation is over, now, students, I have to look at the host now. I have to look at the host. Okay. Let's look at the host now. Now there are three possibilities here. Three possibilities in the host side. What are three possibilities? This, let's say in host A, host B, and host C. Three hosts are here. In host A, the transformation did not happen only. So can I call it as non-transformant? Can I call it non-transformant? Because transformation did not happen on here only. Our plasmid did not go here at all. This is non-transformant. Non-transformant. Our DNA is not present here only. Not a plasmid is not present here at all. Non-transformant. Okay. Non-transformant. Okay. Okay. Non-transformant. Okay. Now, students, these two are transformant, right? But here, in this case, our plasmid is there. In both of these cases, plasmid is there. So can I call these two as transformant? Transformant. I will call them transformant. Okay. But the situation is here I have in this case, non-recombinant DNA has gone. Because there will be many DNA will be there, no? There will be many DNA, right? Non-recombinant plasmid has gone. Directly plasmid has gone without our gene of interest here. But here what happened? This is recombinant. This is recombinant. Or here the plasmid is gone. Plasmid has entered with the right gene of interest. Right gene of interest has gone to the host C. Okay, it has gone to the host C. Transformant, non-transformant, transformant. But in this case, non-recombinant plasmid, here recombinant plasmid has gone. That is with the gene of interest. Now make a small table. I'll make you understand this entire thing. What exactly do we do here? Okay. Host A, host B, host C. Okay. Now here we have, let's take it as ampicillin resistance gene and tetracycline resistance gene. Ampicillin, tetracycline. Now students, tell me, can the host A survive in ampicillin? Tell me. Can the host A survive in ampicillin? Yes or no? Tell me in the chat quickly. Can it survive in ampicillin? It does not have the plasmid at all. It does not have plasmid. It means it does not have the tetracycline resistance gene. It does not have the tetracycline resistance gene. It does not have the tetracycline resistance gene also. Both are not present here. Both are not present here. Okay. See, this is the plasmid. This plasmid is completely missing. This plasmid is completely missing in non-transformant. So it does not have any plasmid. It does not have any plasmid. Ampicillin or tetracycline, both are not present here. Not present here. Now, host B, students, host B. Now, can the host B survive in ampicillin? Yes, it can. It has the ampicillin resistance gene. It can survive. Okay. Can this one survive in tetracycline now? Because it has both tetracycline and ampicillin, both are there. It can survive. It can survive. Last category, in C, students, tell me. Can it survive in ampicillin? It can survive in ampicillin. It can survive in ampicillin. Can it survive in tetracycline, students? Tell me in the chat. Can it survive in tetracycline? All of you? Tell me, can this particular recombinant host C, can it survive in tetracycline? Absolutely not. It cannot survive. So, what did we find here, students? We found here that host C has our gene of interest. It has a gene of interest and it cannot survive in, it is a recombinant DNA take-up. Now, this entire process is called as insertional inactivation. What do you call that? Insertional inactivation. We are inserting something here. We are inserting something here and inactivating this entire thing. Inactivating this entire thing. Now, students, your own question might come to your mind right now. One question might come to your mind right now. If you are a little bit concerned about your entire experiment, sir, all this is okay. I got to know A is non-transformant, B is non-recombinant, and I got to know C, right? C is recombinant. I got to know C is recombinant. It means that it is not able to survive in tetracycline and it is able to survive in ampicillin. So this is my recombinant. Now one question might come to all of your mind. So this is my recombinant, but it is already, but you're in order to find it, in order to find my recombinant, I'm killing my recombinant here. Is that coming to your mind, students? Is that coming to your mind? I am finding my recombinant here, telling that it is able to grow in ampicillin, not able to grow in tetracycline. I got to know C is my recombinant. But this, in that entire process of finding it, I am actually killing my host C. I'm killing my host C. Then what is the use? What is the entire use of, isolation of DNA? I did isolation. I did my desirable gene I found out. After I cut the DNA by using DNA ligase, I cut it. After cutting it, I did gel electrophoresis. After using electrophoresis, I did transformation. After transformation, all of it's dying. So what is the use? How do I even make utilization of it? The answer is very simple. Because every single time we do this experiment in lab, we don't just take one culture, we make multiple replicates, triplicate, quadruplet or pentaplate. So we use multiple copies of this entire research. So just because this batch is dying, but I got to know recombinant is present here. So I will use the same batch itself, I will use the other set. I will use the other copy of it. Simple as that. So do you realize that? So in order to find this recombinant, we have to do some sacrifice. We have to do some sacrifice, but it is worth it in the end. Okay, all of you understood? Now students, now a question to all of you. Okay, now a question to all of you students. Don't you think so this entire process is very cumbersome? Too much of extra work no here? Too much of extra work, right? So who wants to do so much of work? Do you want to know an easier method? Students, do you want to know an easier method? This is very complex, no? So basically you're taking your celebrity, cutting here, inserting it, inactivating tetracycline. Then after that you're growing it into a non-transformant, growing it in ampicillin, growing it tetracycline, right? Making it recombinant. This entire thing looks very complex to me, like very complex. Now you might ask me, sir, is there any easier method? Yes, there is. Now what is the easier method? Understand. What is the easier method? Understand. There is a easier method. There is a easier method. Okay. Now easier method is done in one more type of plasmid called as PUC8. It is called as PUC8. This is your PUC8. Okay. This is called as PUC8. Now what exactly happens here? In this PUC8, here we have a, in this PUC8, right here, we have a ampicillin resistance gene. Apart from ampicillin resistance gene, we also have something called as, LAG-Z gene. Students, tell me how many of you don't understand this concept? Or how many of you feel this is difficult to understand? Raise your hands. Because in next 5 minutes, I'll make it easy for you. That is my challenge. My challenge from my side to your side. In just 5 minutes, I'm going to make it very easy for you. Can we start? Students? Can we start? Yes or no? All of you, tell me the check quickly. Very easy this is. Okay, very easy. Now, understand what happens. What does this LAG-Z gene does? This LAG-Z gene, students. LAG-Z gene generally produces something called as beta galactosidase. It produces beta galactosidase. To this beta galactosidase, if I am adding a chromogenic substance called as X-gal, to this beta, if I am adding the X-gal, okay, if I am adding X-gal to this beta galactosidase, it is producing blue colonies. It is producing blue colonies. Okay, so here we have LAG-Z. In normal condition, LAG-Z gene is producing beta galactosidase. When you add X-gal to it, it becomes blue in color. Okay. Now listen, what happens? Now in case 2, let's take case 2. Now here I have my PUC8, okay. Now here I have my ampicillin resistance gene. Now here I have my lag Z gene, okay. Now what am I doing is, in this lag Z gene is there, right? There are multiple restriction sites there, or recognition sites, multiple recognition sites for different types of restriction enzymes. So what am I doing is, I am cutting this part, cut, and this cut region I am adding the gene of interest here. I am adding the gene of interest at this lag Z gene part. So gene of interest is added to the lag Z gene part here. It has been added here. Okay, it has been added here. Now understand. Now I am adding the gene of interest or alien DNA. I am adding alien DNA here. Now students, once if I add the alien DNA, once if I am inserting the alien DNA, can I tell this lag Z gene is no more? Unfortunately. So if there is no lag Z gene, there is no beta galactosidase. There is no beta galactosidase. It is completely missing. If there is no beta galactosidase, even if I am adding xgal, if I am adding xgal to it, will there be any change in color now? Because there is no beta galactosidase, what will even xgal do? Nothing. So this will become what? White color. This will become white color. It will become white color. So what do you understand? So what do you understand from this simple experiment? We understand that, students, if a bacteria is transformant or recombinant, okay, transformant to be precise. If a bacteria is taking up, how do I know? If I want to know how it's taking up, let's take recombinant here. Since it's bacteria not transformant, since they're using still the plasmid, let's use recombinant. A recombinant one will always produce which color, students, tell me? A recombinant will produce which color? A recombinant will produce white color. Because I know if I'm adding my gene of interest, this is becoming inactivated. Insertional inactivation. This lag Z gene is becoming inactivated and we are getting white color. Now what about non-recombinant? What about non-recombinant? Non-recombinant is going to be blue color. It's going to be blue color here. It is going to be blue color. So non-recombinant is going to be blue color. Recombinant is going to be white color in nature. So students, when you're trying to grow in a media, if you're trying to grow it in a media, this is how it looks. Blue and white. Okay. It's also called as blue and white screening. Now, once you grow blue and white, which one do you take, students? Do you take blue or white here for the next step for transformation? This is before transformation. Right? That's why I wrote recombinant there. That's why I wrote recombinant or transformant. Technically recombinant only. So you take the white one here. Because blue is non-recombinant. So you take the white one and give it to bacteria. So entire thing is going to give white. So white is better. Blue, no. Okay, blue is no. You take white. Okay, white is always recombinant. Recombinant is white. Okay, understood, all of you? Understood? Yes or no? Tell me. See, recombinant is always white. It means that in recombinant my gene of interest is there. Gene of interest is inserted here. So if I take blue, what is the use? Then? Blue is non-recombinant already. There's no point in taking blue. So if I take white, at least, everything will be recombinant and I can make a use of it. All of you understood? Blue and white screening? Students, quickly smash the like button. All of you quickly smash the like button, otherwise The target is not done then. Understood all of you? Isn't it very simple to understand? It's very simple. See here. This is the entire PUC8 gene of interest. Lag Z gene is broken up. Gene of interest is added. Okay. So no lag Z, no beta galactosidase, white colonies. So recombinant will produce white colonies. It will produce white colonies. So white are the recombinant colonies. So a small question here, students, quickly solve the question. Hint 2, I told you, hint 2, always cut DNA molecule at a particular point for recognition sequence. It is how many base pair? I told you already. How many base pair, students, tell me? It is around 6 base pair. I told you already. Okay, 6 base pair. Okay. Next question. The TI, students, these are NEET 2024 question. Not easy question. They are NEET 2024 question. Okay. See this question, all of you. See this question. The TI plasmid of Agrobacterium tumefaciens stands for. What is this meaning of TI? TI is tumor-inducing plasmid. It is tumor-inducing plasmid. Now here the question is, the following diagram shows restriction sites in E. coli cloning vector P/3/2. Find the X and Y. Guess. X is ORI, Y is ROP, right? So ROI, repressor of protein or repressor of plasmid, right? Helps in involvation of relation plasmid. X is responsible for controlling the copy number and link to DNA. So 1 is the answer. 1 is the answer. Okay, 1 is the answer. Next question. Which of the following is not a selectable marker? See, ampicillin can be selectable marker. Chloramphenicol can be selectable marker. Tetracycline can be selectable marker, but not metformin. Not metformin. Okay. Look at this question, students. Look at this question. This also is NEET PYQ. Ampicillin is selectable marker. Okay, A is 3. A is 3. ORI site where replication starts. Multiple cloning sites is restriction sites where we can add use restriction. Beta galactosidase is part of blue-white screening. So A is going to be 3, B is going to be 4, C is going to be 1, and D is going to be 2. So answer is 1. Answer is 1. Restriction endonuclease find its specific recognition sequence and bind to the DNA. Yes, it will go scan the entire thing, it will bind there. It will bind there. Restriction endonucleases cut each of its 2 strands of the double helix at a specific point in their sugar phosphate backbone. True. Both are true. NEET 2024 question. In gene gun method used to introduce alien DNA into host cell, microparticles of dash metals are used. Tell me the answer in the chat quickly, all of you. All of you tell me the answer in the chat. Microparticles of which particular metals are used? Inert metals. Inert metals, students. Inert metals. Tungsten and gold. Tungsten and gold. Answer is not given, but let me write the answer for you. Tungsten and gold. Tungsten and gold, students. So students, with that, we come towards the 70% of chapter is over now. Let me tell you, students, the entire chapter is going to be over in the next 20 to 25 minutes. So for the next 20 to 25 minutes, are you ready for a power pack session? Yes? Ready, all of you? After that, we'll be doing applications. Gopika ma'am is going to come, and then Gopika ma'am will start after that. Okay? Can we start, all of you? Can we start? Next thing is going to be the entire process in rDNA technology. In the next 20 to 25 minutes, if you stay focused, you will finish the entire chapter. You will be taking home those juicy 38 marks, right, to yours. Okay? Can we start, all of you? Yes? Let's start with the first concept. Okay? Now, students, recombinant DNA technology involves several steps in specific sequence, such as this is the actual sequence which has to undergo. Actual sequence. First step is called as isolation of genetic material. First step. Second step, cutting of DNA at specific locations. Students, tell me, how do you cut the DNA molecule? How do we cut the DNA? With the help of the chop, 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 chop. What is that? Restriction enzymes. Okay. Next thing, amplification of gene of interest, that is PCR. Insertion of recombinant DNA into the host, that is basically the transformation. Then obtaining the foreign genetic product, gene product, we get the product from there. And finally, downstream process. I'll tell you what is downstream process, so don't worry. Now students, I'll teach you how to isolate DNA at home also. You can isolate the DNA at home also. It is very, very, very easy. Okay, very easy. Now, first one is called as isolation of DNA. Now students, if you want to isolate the DNA, first of all, if you want to isolate the DNA, you have to remove the outer barriers. When I say outer barrier, I mean the cell membrane as well as the plasma membrane, as well as cell membrane plus the same, or the cell wall. Okay. For example, in the case of bacterial cell, there is a cell wall is there. Plant cell, the cell wall is there. Fungal cell, there is cell wall. But in all of these cases, in bacterial cell, if you use lysozymes, it can completely break the outer layer. In the case of plants, cellulase, chitinase in the case of fungal cell. So if you do that, the entire outer layer is going to be broken down. Now the next layer is called as the cell membrane or the plasma membrane. The entire plasma membrane can be broken down if you use something called as a detergent. Soap also is possible. You can use a normal soap also. So if you add a detergent, entire outer layer is broken down. Now once that outer layer is broken down, we have to remove the unwanted things from the inside. For example, there is protein, there is RNases, and there is these two things. Now how do I remove the protein? And there is also lipids somewhere. Lipids are also there. Now for lipids, I will use lipases. For proteins, I will use proteinases. For RNA, I will use RNases. Now, should I also use something called as DNases? Students, tell me in the chat. Should I use DNases? Absolutely not. If I use DNases, the entire thing will be useless. Okay, so don't use DNases. Keep the DNA. So once you remove one by one, everything will be used. Now once entire thing, even RNA, everything is removed, only the DNA molecule is remaining. Now this DNA molecule has to be precipitated out. How do you do that? For precipitation, you add chilled ethanol. If you add chilled ethanol, and the entire DNA will get precipitated. Can you see here? Entire DNA will get precipitated. Okay, see here. This is addition of chilled ethanol. If you add chilled ethanol, the entire DNA is getting precipitated. Now after that, there is a step called as spooling. Now if you use a small spool-like structure or a small needle kind of thing, you can remove the DNA from it. You can easily remove the DNA from it with the help of called as spooling. Okay. Now in general, you don't have to actually do the spooling. There are multiple alternatives to it. Let me do the alternative. Spooling is not required. Okay. Now, what you can do is, chilled ethanol plus cell is there. Add it to a centrifuge. Completely centrifuge it. Simple. Instead of you physically doing the spooling, you can simply do the centrifuge also. Once you centrifuge, because of change in gravity and the weight and everything, right, all the supernatant will come out. That is the debris which we don't need. And in the bottom there is a pellet. Pellet will have the pure DNA. So pure DNA will be found in the pellet, which can be used for the next step. What are the next steps, students? Next step is called as cutting. How do you cut, students? We cut with the help of resection endonucleases. After cutting, we can do separation. Separation with the help of gel electrophoresis. Okay. Now, students, next step is amplification. Next step is something called as amplification. Very simple process. Listen to me. Amplification means making multiple copies of the DNA. Now, how do you make multiple copies? The multiple copies are made using a simple technique called as PCR method. By PCR method. This entire method of PCR was developed by Carymulus. Okay, developed by Carymulus. This is the entire PCR. PCR is called as polymerase chain reaction. Okay, here we take a piece of DNA and amplify it within a lab. That is why it's called as in vitro condition. In vitro. Okay. So in vivo condition. Inside the lab, right? Now, how do we do it? Now, for example, students, I tried to find the image, but I could not find the image. There was an image of me working with PCR, but I could not find it. If I find it, I will show it to you. Okay. I still remember I used to sit hours together, 4 hours, 5 hours in front of the in the case of thermocycler, in front of the thermocycler. This is the thermocycler. Can you see? It's called as your PCR machine also. It is called as PCR machine. This thermocycler can be used to make multiple copies. Okay, thermocycler. Now, how do we get before you add into the DNA, before adding the DNA to make multiple copies, you need to prepare the entire solution first. In that solution, we need to first of all, you have to add the DNA, obviously. Apart from DNA, we should also add the primer. Now, you might ask me, sir, why primer? Students, you tell me, in DNA replication, do we need primer? Can the replication of DNA happens without the primer? Forward primer, reverse primer? Not possible. So if you want the replication to happen, we need primers. Apart from primers, we also need DNTPs, that is deoxyribonucleotide triphosphates, to make up the entire thing. We also need the DNA polymerase. Now students, here is a small twist here. In the normal replication, right, in the normal replication, we have the normal DNA polymerase. But here, this entire reaction happens in the very high temperature, and we cannot use the normal primers, normal polymerase. Because of that high temperature requirement, here we have a primer, so here we have a polymerase which can work at high temperature. That is called as Taq polymerase, Taq DNA polymerase. And this Taq DNA polymerase is specially extracted from a bacteria called as Thermus aquaticus, from Thermus aquaticus. Okay. So here we have the gene of interest, 2 primers, DNTPs, Taq polymerase, Thermus aquaticus. From here we get it is present in hot springs and everything. So this polymer can even work at high temperatures. So once we have all that, add into a vial, keep it inside a thermocycler. Okay. Temperature, duration set. Now with that, your entire process started. Entire PCR is started. Now once PCR is happening, nicely it is mixing. There are 3 main steps here which undergoes. First step is called as denaturation. Second step is called as annealing. Third step is called as extension. 3 main steps in PCR. Now what is the meaning of denaturation? Because of high temperature, it's around 94 to 96 degrees Celsius. Because of this high temperature, the entire genetic material will become from double stranded to single stranded. Okay, you see, single stranded. Denaturation. After denaturation, next step is called annealing. In annealing, we add the primers. This primer, forward primer, reverse primer, both are added. This entire thing mainly works at 55 to 60 degrees Celsius. Okay. Now, next step is called as extension. Now, using the DNTPs from 5' to 3', extension will happen. Always remember, 5' to 3'. This will roughly work around 70 degrees Celsius, 72 degrees centigrade. You see, it will keep on extending. And that is how from 1 copy we got 2 copies. 1 copy to 2 copies. Okay. See here. Polymerase interaction. 1 cycle we get 2 copies, 2 cycle 4 copies, 3 cycle multiple copies, right? Keep on getting the copies. There is a simple formula here. 2 to the power of n is equal to number of copies. Okay. So if let's say 2 to the power of 3, third cycle, tell me what is it? That is nothing but 2 into 2 into 2. 2 to the 4, 4 to the 8. So we'll get 8 copies. In 3 cycles, we'll get 8 copies. Okay. Now, after amplification, we have to insert it. After amplification, right, we have to insert it somewhere. Inserting. Can you see? And how do you insert, students? How do you insert? Gene gun, holistic method. We learned already micro injection, gene gun, heat shock method. All of that can be used for inserting the plasmid into the host. Okay. So vector DNA, plasmid, gene of interest, rDNA. This is the recombinant. This is now recombinant rDNA or recombinant DNA. Adding it to the host, transformation. And this becomes transformant also, transformant. Okay. Now finally, inside the host, our desired product is being produced. For example, if it is insulin, insulin is produced there. Insulin is produced there. Insulin protein. Okay. Now students, after this entire process, it takes months together. It takes months together to do this. After this entire thing, right? After this entire thing, why do we need to large scale it? Like, is it required to do large scale? Yes, it is required. It is required to do the entire thing at large scale. Obviously, not enough produce because microbes are producing only this much. Not enough produce. Second thing is tedious process which requires monitoring, not cost effective. Not cost effective at all. So this takes a lot of time. So you have to large scale it. Now, how do we upscale this? One small thing I'll ask you students. Listen to me. Karthik, tell me, when your mother is cooking in the house for 4 people or 5 people, do you use small cooker? Now, suddenly, if uninvited guests come to your house, some random relative who you never spoke after the childhood, who tells you, oh, you're grown so big and everything, okay, random relative, he comes to your house with his own family and everything. Can we still cook the food in the same small cooker? Not possible. Not possible. So what do? We take the big one, the entire tumbler, which used in weddings. In weddings they use, right? That one you pull out. So such thing is also there. Instead of using a small cooker in the house, we use a large tumblers, and that large tumbler is also called as bioreactor. It is called as bioreactor. Okay. So before you go to bioreactor, you need to understand what is upstreaming and downstreaming. In upstreaming is the entire chapter. Upstreaming is the entire chapter. Isolation of desirable gene, cutting it, pasting it inside the plasmid, making the plasmid competent, recombinant plasmid, taking the plasmid, adding it to the host. This entire steps we learned in this entire chapter, right? That is part of upstreaming. Then what is downstreaming, students? Understand. Upstream, we learned, right? Selection of desirable, upscaling, bioreactors. The last step is bioreactor here. Okay. Culture, storing it, culture broth, upscaling. This is a bioreactor. Can you see? Bioreactors are basically large cooker. Okay, large cooker. Can you see? Large cooker. In this large cooker, once you add the entire product, it'll keep on making the, if you add the bacteria, if you add the nutrients, it'll keep on making the multiplication. Bacteria will keep on multiplying inside. Okay, keep on multiplying inside. Okay, so raw materials are basically proteins, enzymes, vitamins. Basically everything required for the bacteria to grow nicely. Okay. Cells can be used, plants and animals can be used to the host. It can provide proper temperature, pH, substrate, salts, vitamins, everything is maintained, students. Every single detail is mentioned here, maintained here. Okay. Can you see this is called as stirred-tank bioreactor, which is the most famous one, most preferred one also, most preferred one. In the stirred-tank bioreactor, can you see this sample port where the sample is added inside? This is the entire thing is called a reactor tank where the entire multiplication happens to the bacteria. Can you see temperature control here is there? Aeration or sparge where you can release the oxygen for aerobic bacteria. Yes. Form breaker, because when the entire reaction is happening, there is a formation here. The entire form is broken down. It is broken down. Okay. Now there is single batch and fed batch. Now what is the difference? In single batch, this is still tank single batch. In single batch, once you add all the requirements and you close it, and until the entire reaction is over, you can't open it back. Right? So it is closed system. Nutrients and culture provides only once if you add, everything is enough here. Right? Now here bacteria, understand. Here bacteria is present in lag phase. Bacteria will grow here nicely by taking all the nutrients, and eventually all the bacteria will die. Why? Because nutrients are over, bacteria is dying. Okay, that is what happens in the normal closed or single batch bioreactor. Now students, there is something called as continuous bioreactor. Okay, continuous. In continuous bioreactor, can you see? There's something called as feeding pump. Because the feeding pump, we keep on adding the nutrients. Now students, you tell me, if you keep on adding the nutrients, after reaching the exponential phase, the bacteria will keep on surviving. There is no death phase here because nutrients are added, bacteria will keep on eating the nutrients, keep on multiplying, and the entire reaction will keep on going without stopping at all, without stopping. Okay, that is called as continuous batch. Okay, a single batch is safe, costly, but time consuming, cost effective, unsafe little bit, and less time it takes. Okay. Now, what is the downstreaming process? Understand. So, upstreaming process is the entire hard work which you do. But if you don't do downstreaming process, right? If you don't do proper downstreaming process, the entire work is useless. Why? Because if you're making something product, right, we want consumers to take it. For example, let's take I am only there. In my MSc, I did some research. I worked on a plant called as Samia furfuraceae. I'm not making up, this is my actual research. In that research I found that my plant, that is the first time in the world, no one has done it on it. No one has worked on it, this plant called as Samia furfuraceae. It is a gymnosperm. I found that this plant is able to produce antibacterial and antimicrobial, antibacterial and antifungal properties. I found that, I actually did. Okay. Now that was my upstreaming process. That was my upstreaming process done by me. But I did not have enough money, and obviously my MSc was over, my post-graduation was over. I did not do downstreaming. So whatever work which I did, apart from getting marks for it, did I get any money out of it? No, because I did not even sell my product. I did not sell my product. I just kept my research. It is still there. I can actually sell my research to someone also who wants to do it in future. But I did not do downstreaming. Now, what is the meaning of downstreaming now? What is the exact meaning of downstreaming? Understand. And how can you make money here? In downstreaming, there is something called as recovery and purification of formed product. Because I made one product. In my MSc, I made a product. But I did not do purification. I did not make it more pure for consumption. Do you realize that? I can make a product, but it has to be the purest form if someone wants to consume it. Okay. Okay. Removal of undesirable elements found by the cells present in the culture media. So removal of undesirable elements I did not do. So basically what happens is separation of undesirable things, disintegration of cells, extraction and concentration, purification. So the entire downstreaming process involves much more pure nature of your product. I did not do that. Okay. Pure nature. Now you have to do clinical trials, obviously. This also was requirement. For example, I made one, let's take one tablet which is 1000 times more effective. Let's take a quick example. I did not do this, but quick example. Let's take, I have a paracetamol, which I knew paracetamol I made. I will call it paracetamol neo. I will call it as paracetamol neo. Okay. Now this paracetamol neo is, let's say it is 10 times more effective than normal paracetamol. 10 times more effective than normal paracetamol. But a normal paracetamol, let's say, a normal paracetamol is 2 rupees. 2 rupees. Mine is neo paracetamol, new one. Neo one, I am selling it for, let's say, 200 rupees. Even though mine is 10 times better. This paracetamol is 10 times better, but the cost is 200. Now you tell me, students, all of your consumers, which one will you buy? Will you go for 200 rupees one or will you go for 2 rupees one? Tell me, which one will you go? Tell me. Tell me. Which one will you go for? Tell me. Which one will you go for? Will you go for 200 rupees one or 5? Don't tell lies. Just to muska you don't do. Okay. If I was your rules, I would go for this 2 rupees one because cost effective. Even though this might work 10 times better, I will still not go for it. Okay. So you should always make sure that it is comparative. It is comparatively efficient. Okay. Proof of concept. That is, you have to do testing in many public, right? And dosage. This is all part of downstreaming process. Okay. Quality control should be done. Check for adulteration. Checking for packaging. Everything has to be done. That is all part of downstreaming process. Downstreaming process. Okay. Tell me the answer to this question, students. Tell me. Which one is the answer here? Which statement is incorrect? Which statement is incorrect? Most commonly used bioreactor are stereotype. I told you, yes, correct. Bioreactors are used for production of small scale, wrong. Bioreactors have agitated system, oxygen delivery is yes. This is an easy question. This is an easy question. During purification process of recombinant DNA technology, addition of chilled ethanol precipitates. So students, it was 23 question. Such a laddu question this is. Such a laddu question this is. Such a laddu question. Precipitate DNA. Upon exposing UV radiation, DNA stained with ethidium bromide will show. It will show bright orange color. Bright orange color. It will show bright orange color. Okay. And students, with that, right? With that, chapter is over. With that, in just 2.5 hours, do you believe that? In just 2.5 hours, the chapter is over. Just like that. Just like that, chapter is over. And students, tell me, right? Tell me, did you understand every single concept? Starting from how much I am bro concept. Starting from what is traditional. Starting from what is traditional. What is modern biotech. Starting from how you can make curd, dosa to vaccine and antibiotics. Take a small recap. See here. Starting from. Then we learned about modern biotechnology. Right? Then we learned about this entire concept of genetic engineering and bioprocess engineering. Genetic engineering is entire isolation. Bioprocess is sterile conditions. Yes or no? Then we learned about the entire concept of Bro and Macha, where we isolate DNA, that is isolation of gene of interest. Then we add it to our vector, that is basically addition to the vector, right? It became recombinant. This one is recombinant now. Recombinant. Then we learned about transformation. Host will grow. It will grow. Okay. Now. Then we learned about RNA technology, how it was introduced with the help of the Stanley and Herbert. Then we learned about steps, tools, restriction enzymes, ligase, vector, host organisms. Then we learned about the entire restriction endonucleases, where we can cut in the sticky ends or hanging ends or blunt ends or flush ends, which we get here. Right? We learned about palindrome sequence and everything. Then we learned about this entire thing. Then we learned about nomenclature, how nomenclature plays a role here. Then we learned about electrophoresis. DNA being negative charge, it goes towards the anode. Small things, small DNA goes further. UV transfiltrator. There. UV transfiltrator we can use. Transfiltrator. Then we learned about vectors, cloning vector. A vector can do help in carry the gene of interest into the host, and also cloning. Why cloning? Because of the ori site. Because of ori site. Different type of vectors. Plasmid, phages, Bacterial. Okay. Bacterial. Then we learned about a celebrity, PBR22. We learned about ampicillin, tetracycline. What is present in each? What is ori site? Right? Then we learned about how to make a competent by using a divalent cation, so we can make the entire thing competent. Then what is a transformant? How can we do transformation? Then we learned about heat shock, microinjection, ballistics, and disarm pathogens. Then we learned about insertional inactivation. This is for the using for the celebrity PBR22. Then we learned about the laxate gene. Done. Blue and white screening. Then we learned about DNA isolation, PCR method, and downstream process. So with that, your entire chapter is over. Now students, if you go, right? If you go to the description of this video, you will be able to find questions, right? You can solve questions. Let me show it to you. Let me show it to you. I hope it is visible. No, when you add the DNA, right? Sir, whether you put the DNA after melting the agarose gel in it or pouring the tray into the process separation, no. What do we do is we generally take the piece of after agarose gel is there, right? We make the gel, it is hot itself, it should be solid. When we are adding the DNA into the agarose gel, it has to be solid, it has to be jelly like. Okay, if it is not jelly like, it will melt into it. So we melt in the starting, but we after it is liquid, we pour the liquid into the tray, but we wait for the tray to cool down. We wait for the agarose gel in the tray to cool down. Once it is cooled down, then we add the DNA. Then we load the DNA. Okay, then we load the DNA. Students, all of you quickly smash the like button. All of you quickly smash the like button. Make it. It's not 200 return. Scam. Students, if you go here, okay, I have to change it. We'll get it changed. Don't worry. Description of video. If you go, you'll be able to find it. Okay. Now, students, Gopika ma'am will come now. Right. Gopika ma'am will come, and we'll be starting with the application right now. So everyone, read out in the chat for Gopika ma'am. I think there will be a break right now. I think all of you need a break, right? All of you need a break. Okay, break until 8 p.m. Okay, break till 8 p.m. Let me just give a small break. Okay, so at 8 p.m. application will start. Okay, so students, ma'am will see you. Ma'am will meet you at 8 p.m. So until then, wait. Okay, don't run anywhere.
Hello students, welcome to Vedantu NEET English. Am I audible and visible to all of you? So sorry, few minutes delay happened. So welcome all of you to Vedantu NEET English. I am Ms. Gopi, Biology Master Teacher. So are we ready for biotechnology applications? Because now you study principles, you need to know where do I use these principles. I have studied so much about recombinant DNA technology, inserting a gene, getting my desired characters, but exactly how is it going to benefit? How is it going to benefit everyone or anyone, right? Ready all of you? Awesome. Okay, likes are a little less, but it's okay. On the go we should be able to make the likes. Okay, so we will have a fast and furious session as well, because biotechnology applications is a very cutie chapter, so we will try to finish the chapter as soon as possible. Okay, ready, study, let's go. Okay, so students, biotechnology applications, as I told you, whatever you studied in your principles, all that things we are going to use here. Now exactly, ma'am, where do I use all these applications? First of all, you will use it for agriculture. That is the main place that you are going to utilize all of this. Okay, that is agriculture. All of you have heard about organic farming, right? Organic farming. Ma'am, nowadays I eat only organic apple. What do you mean by this organic farming? Where they are using your pesticides, fertilizers, everything is in organic mode. That means less of chemicals, more of, things that will not harm our body, right? This is basically what you mean by organic in a very layman's language, if I'm trying to say. Now, we use this organic farming. At the same time, in agriculture, people have come up with GMO, which is called as genetically modified organisms as well. Okay, and where we have introduced also transgenic animals. At the same time, we have also used agrochemicals. Okay, agrochemicals, and we also did green revolution. So students, I'm just telling you the entire chapter in just one summary types. Okay, just the introduction. This is, we will go into depth of each of the topics. Clear, all of you? Yes. Oh, 20 more likes. Okay, all of you, quickly smash the like button. Okay, clear. Is it reflecting too much? 1 second, students. Okay, now it's better. Yeah. So, agrochemicals, green revolution, genetically modified organism. We also spoke about not just in agriculture, but we come to us, like in organisms also, we speak about Bt cotton. We speak about how we can do gene therapy, how we can do gene subtraction, how we can do gene addition. So the entire principles that you studied about recombinant DNA technology, you are going to use that, or you're going to apply this today in your day-to-day activities or in your real life. Okay, real life, not real life, real life, where and all you can use these things. Okay, so this is just the introduction about how you can use this. Students, most of this chapter I will be teaching you with NCERT lines, because major chunk in this chapter is just theory. Okay, wherever it is written, okay, or wherever I need to make the notes, I will be giving the notes to you, don't worry. But major things will be NCERT based. Okay, so look here, what is the whole application about? Providing the best catalyst in the form of improved organism, usually a microbe or a pure enzyme. Okay, pure enzyme, creating optimal conditions through engineering for a catalyst to act. So basically, biotechnology means what? You do some kind of modification, and that modification benefits. Okay, that modification ultimately benefits something, right? Now, downstream processing technologies to purify protein and organic compounds. And this is one of the topics that you studied last in your principles, right? All of you tell me in the chat box, what did you study? Is my voice breaking, or is it only for Sanjana? Tell me in the chats. Voice is fine? Please tell me. Is there any issue with the voice or any other problem? Okay. So look here, students, downstream processing means what? Purifying. Okay, purification. We have to see the authenticity of anything that I'm taking to market, right? Today, let's say I'm making some medicine. Now, how do I know this medicine is going to be commercially used or not? Until I go through an entire downstream processing where I do the entire purification, okay? Entire purification. Just now, Basava sir taught you, right? Just now, Basava sir taught you, so it should be in your head right now, fresh. I think all of you haven't shared the link with your friends. Why? Biology 360 Club is enough. Josh is less. Josh is less means club also will get shut down, okay? If you people don't come, then why will we open the club and sit? So yeah, so energy shouldn't be less, and you should keep calling your friends also. Now students, two topics have been added from your strategies to this chapter, that is tissue culture and your somatic hybridization. So the entire micropropagation, tissue culture, somatic hybridization I will teach you in just one go, because every topic is connected. Okay, every topic is connected. Okay, hi ma'am, I am Priya. So what should I do, Priya? Okay, so tissue culture, somatic hybridization has come from strategy. So I will start with tissue culture. Now students, if I want to do the entire thing in an in vitro condition, that is exactly tissue culture. Ma'am, what do you mean by everything? So in tissue culture, students, what I'm going to take, I'm going to take a small portion. Okay, it could be a cell. Okay, it could be a tissue. It could be a cell. It could be a tissue. Okay, it could be a meristem. I can take any of this and all this, any one small portion of a plant if I take, okay, very small portion of a plant if I take, and I use it, I will call it what? I will call it an explant. I will call it an explant. Now this explant under certain conditions, that means under sterile conditions, okay, everything has to be sterile. Under sterile conditions, I will be allowing this to grow. This will grow into a mass of cell. This will grow into what, students? A mass of cell, which is called callus. Okay, mass of cell. And this will later grow into a completely new individual. Okay. Now, what is going to happen? This explant, okay, this explant shows a characteristic. What is that characteristic called? All of you tell me in the chat box. Totipotency. What does it show? Totipotency. Totipotency is the ability of a part of the plant or a cell to completely grow into a new individual. Okay, ability of a cell to grow into a complete new individual. Clear to all of you? This explant has the ability to grow into a completely new plant. Understood? Yes or no? Yes. Okay. Now look here. This explant shows totipotency. Now you will ask me a question, ma'am, how did this exactly happen in the in vitro condition? So let's take a Petri plate, students. Okay, this is a Petri plate. In this Petri plate, let's say Meghana put an explant. Okay, Meghana put an explant. It could be a meristem, it could be a cell, it could be a tissue, anything of it. Okay, any sort of one small portion of a plant we put. To this, I am going to provide a nutrient medium. I'm going to provide what? A nutrient medium. Because I know that I'm growing it in lab, and Meghana has given me just an explant. Now in this Petri plate, I have to give whatever is required for this plant to grow. Now what example, like what do I exactly give? I will give nutrient medium, students. First I am going to give a lot of carbon source. I'm going to provide carbon source like your sucrose. Okay. Now after that I'm going to give inorganic salts. I'm going to give some vitamins, some amino acids. Okay, these are few things that I will be giving the plant. With that, I'm also going to give some hormones like auxins and cytokinin. These are exact lines of your NCERT. Okay, so make a note. So Meghana gave me a small portion of an explant. To that, she told me, ma'am, please add whatever you think is going to grow this explant to a completely new plant, because I cannot do this outside, because I will not get that much numbers. So you give me a solution that you do this in the lab. So tissue culture is a system, is an entire process where we take a small portion of the plant and grow it in the lab. And I can grow multiple numbers. Okay, how much ever numbers I want, I will grow it in the lab with each conical flask having the nutrient medium. Is it clear to all of you? Thumbs up in the chat box. I'm very disappointed in the crowd, students. I don't know where all of them went. This is why we don't give breaks. Okay, let me just tell them. Yes, okay. Tell me. Okay, good. Some of them came now running, saying that, oh my god, ma'am is scolding. Okay. Okay, very good. So I did what, Meghna told me. Now I also gave growth regulators. Okay, now the thing is, students, in a very shorter period of time, okay, what is the use of this? In short duration, I'll be able to get multiple or many plants, right? I'll be able to form many plants. And these all the plants are going to be genetically identical. Okay, genetically identical. For genetically identical plants, we will call them soma clones. Clones means what? Now I give you a Xerox paper, right? I give you a Xerox paper, you went to take Xerox and you took the Xerox and came. That means what? They're all identical to each other. Clones. Okay, so genetically, okay, I'm writing it here in a asterisk mark. Genetically identical, okay, plants, okay, genetically identical soma clones. Okay, soma clones. So understood tissue culture, very small portion taken from the other place and have come. Okay, very good. Yes, Gopika ma'am army, most of them are here because I asked them, where are you? Now, this is clear to all of you. So, students, this entire thing is micro propagation only. A small portion of a plant is taken and they are propagating at maximum amount. In a smaller time, I'm getting maximum number, right? In a smaller time, I'm getting maximum number. Clear to all of you? Yes? Perfect. Okay. So, with this tissue culture, we can grow plants like banana. Okay. Banana with micro propagation, apple. Okay, these are few examples that if you can remember, well and good. Okay, well and good. Yes, and sugar cane. Okay, sugar cane. These are few things that you have to remember. So micropropagation is clear. I've also put NCERT snips, so don't worry. Okay. Now, next most important thing is micropropagation is clear, tissue culture is clear. Now we will learn about somatic hybridization. Okay, we will learn about, I think I've put a slide for somatic hybridization. Yeah, we will learn about somatic hybridization. Now, what is somatic hybridization exactly? That is, students, where we mix two things, okay, and we get something out of it, like tomato. Tomato plus potato is equal to pomato. Now, what is pomato? So, look here. Somatic hybridization means we take only the protoplast of the cell. Let's take, I have variety, okay, I have variety 1 and variety 2, okay, variety 2 I have. Now, from these two varieties, I am only going to take the protoplast. Before I take the protoplast, what I should do? I should be able to digest the cell wall, right? I only need the inside thing. What is inside? I need the plasma, I need the nucleus, that's all I need. I need the inside portion of it. So here what I'm going to do, from these two varieties, we are going to make sure to digest the cell wall. Very good. Someone is telling fusion of protoplasm. Very, very good. So here from this, now I have protoplasm 1, right? Protoplasm 1, and here I have protoplasm 2. Protoplasm 2. Now, taking these two protoplasms, I am going to make a fusion. Okay, I am going to do a fusion. Fusion of protoplasm. Now, all of you tell me, fusion of this protoplasm will give me a new variety. Yes or no? New variety. This whole process is called as somatic hybridization. With this help of somatic cell, okay, we are trying to form a new variety out of this. Like this only pomato would have come. Okay, pomato came. So this entire thing is called as somatic hybridization. Okay, students, small portion added from strategies chapter. I have put the PDF of it, so don't worry, you will be able to get everything in this notes. You can follow me on the Telegram, Biology by Gopika. Okay, clear? Yes, very good. Fusion of protoplasm, that is called as somatic hybridization. Yes, very good. Okay, somatic hybridization. Yes. Now, students, remember, as I told you about somoclones, again, with this somoclones also, we can do one more thing, that is, in this entire process of biotechnology, we can also do one thing, that is, from a deceased plant. From what? From a deceased plant, I can get a healthy plant. From a diseased plant, I can get a healthy plant. Ma'am, how is that possible? Now, let's say this plant is deceased. Okay. Okay, this plant is deceased with virus. Okay. One virus has affected this plant got deceased. But even though this plant is deceased, what is going to happen? I will only take, okay, from this plant, I am only going to take the apical and the axillary meristem. Okay, what am I going to take? I'm going to take the apical or the axillary meristem, and I am going to form a new plant. Okay, I am going to form a new healthy plant. New healthy plant. How is this possible, ma'am? This got affected by virus. Now, students, when virus affects, please understand, in this meristem, these meristems are actively dividing. And also, in your axillary and apical meristem, the plasmodesmata, through which the cytoplasmic exchange can happen, that is a little underdeveloped. So, because of this, the entire plant will not be affected. There will be some cells which I can use as an explant to grow into a completely healthy plant. Is this clear? Okay, so apical meristem and axillary meristem I will take from this deceased plant which was affected by a virus, and from that I will make a new healthy plant. So don't you think biotechnology is very, very helpful? Making this genetically modified organisms also is beneficial because they are disease resistant. I can make high yielding one. I can make the one that I want with the varieties that I want. All this I can do with this, right? So this is also one of the most important things that you have to remember. Any doubts so far? Please let me know in the chat box. Perfect. Disease plant clear? Yes. Chat box only I can see few people, okay, responding. So this is the three major portion, okay. Tissue culture, your disease plant, as well as somatic hybridization. Somatic hybridization is very easy, okay. Just the mix of protoplasm, fusion of protoplasm will give you a new variety, and this will lead to the formation of somatic hybridization, okay. This is somatic hybridization. Now let's go through NCERT. So we understand that, okay, if we have understood every single line of NCERT. See students, as traditional breeding techniques fail to keep the pace with the demand of provide sufficiently fast and efficient system for crop improvement and other technology, we call it tissue culture. That means because of the increase in population, because of the increase in the amount of, number of people out there, what happened? We are not able to increase the resources. Now you think that, okay, in the bro who wanted to become tall in Baswas's class. Let's say in bro's house 5 people are there, and in Macha's house 15 people are there. Now will Nagaraj in this one corner of Karnataka who is making rice or who is growing rice will be able to know that Macha has 15 kids, bro has 5 kids? No, Nagaraj is not bothered. Nagaraj is only growing the crops that he can afford. He can, cultivate in his farmhouse, right? Cultivate in these places, correct? So, understand that when the population increases does not mean resources also have to increase. We try to increase the resources, but it will not be equal to equal. Yes or no? So that is what here happens, students. Because of the increasing population, we started tissue culture. That is where what does tissue culture mean? See here, okay, here, yes. So any part of the plant, okay, any part of a plant taken out and grown in a test tube under sterile condition in special nutrient medium, okay, special nutrient medium, this is called as tissue culture. What is going to happen? There will be racks. Each rack will have conical flask. On top of the conical flask, there will be tube lights kept. Inside this conical flask, the entire nutrient medium will be mixed, which has carbon source, which has your auxins and cytokinins, inorganic salts, vitamins, amino acids, everything. A mix would be made, okay? Mix would be made. This capacity to generate the whole plant from any cell or an explant is called as totipotency. That means the ability or the capacity with which that they can develop into a completely new individual. Clear? Yes? Okay. Clear? Okay, perfect. Now, So total dependency is clear. Now see, it is important to stress here that the nutrient medium must provide a carbon source such as a sucrose, okay, also inorganic salts, vitamins, amino acid and growth regulators like auxins and cytokinins, gibberellins, okay, depending on the situation they will be injecting the hormones as well, okay. Every line of NCERT I have written in that slide. By application of these methods, it is possible to achieve propagation of large number of plants in a very short duration. Very, very important. They can ask you a question, why is tissue culture even there? Okay, why is tissue culture even necessary? So you can write this point, okay. Large number of plants in a very short period of time, you can make it. Now, this method of producing thousands of plants throughout the tissue culture is called as micropropagation. Any doubts in the students? Tissue culture again comes under your micropropagation. Why? Because we are talking about small micro things that can propagate maximum, okay. Micropropagation. Each of these plants will be genetically identical to the original plant, so they can be called as soma clones. They can be called as soma clones, okay. Many important foods like tomato, banana, apple, sugar cane, all of these are a part, okay. Okay, tomato, banana, apple, okay, have been produced on commercial scale using this method. Try to visit a tissue culture laboratory, okay. In our college and all, it was there. That I did not take any photos, or else I would have shared it with you. Now, another important application of the method is the recovery of, very important, healthy plant from diseased plant, okay. Healthy plant from diseased plant. That is, even if the plant is infected with a virus, there are some meristems which will not get affected. Why? Because meristem cells are actively dividing. Because of that, it is not easier for the virus to affect. Second thing, what I told you, plasmodesmata will be underdeveloped, okay, in these meristems. Because of that, mixing or movement will not happen, okay. Movement will not happen. So meristem, that is your apical and axillary, is free of viruses. Hence, one can remove the meristem and grow it in vitro to obtain a virus-free plants, okay. Scientists have succeeded in culturing the meristems like this. Now, scientists have even isolated, look here, single cells from plants, and after digesting their cell walls, this is what I told you, they have been able to isolate naked protoplast surrounded by the plasma membrane. Isolated protoplast from two different varieties of plants, okay, each of which I want desirable characteristics. Let's say Tanuja and Madhumati is there. I want from Madhumati attendance and I want from Tanuja likes. Now what I will do? I will do a mixture of this. I will tell Tanuja, you put the likes, Madhumati, you make the attendance. So what is going to happen, students? This is going to be a combination of my desired character. Same thing like this. Protoplast 1 plant, let's say they are going to give more flowers, and protoplast 2 has, you know, very healthy root system and shoot system. These two I am going to mix. This is called as somatic hybridization, okay. For that, the first step will be digesting the cell wall, okay. Yes, each having a desired characters can be fused to get hybrid protoplast, which can be further grown to form a new plant. So a new variety we formed with the help of somatic hybridization, and this process is called somatic hybrids, okay. So even if a question comes from here, you people don't have to worry, don't have to think about, oh ma'am, I did not know, I don't know what to do and all of that, okay. Don't worry, this is the question, okay. This is the topic and this is the question from where it will come. Now this is called as somatic hybridization, and students, these can be called as somatic hybrids, okay. These can be called as somatic hybrids, okay. Somatic hybrids. Yes, clear to all of you? Now, imagine a situation when a protoplast of tomato, okay, is fused with potato. First of all, this is the full mixture, right? Because fruit, vegetable confusion will be there. And then you grow to form a new hybrid plant combining tomato and potato. So this was done mainly for people who are very lazy to cut tomato and potato. So they were like, oh, let's just make tomato. No, very nice. What an easy thought process. I can just make tomato and I'll make tomato sabji. Next day when I go to, you know, office and school, I'll tell, yeah, I made bhuji. What bhuji? Tomato bhuji. Because I think every time it will not work. Just because you want Amitabh Bachchan's height and Salman Khan's body, you make a mixture, it will not work like that. Okay, it has to make sense, no? Yes. Okay, very nice. Now, very important next topic is Green Revolution. Green Revolution, students, was mainly done Because of the growing population, okay, growing population. So green revolution actually was an entire concept where we had a 3x growth. What growth? 3x growth. They can ask you a direct question telling that green revolution led to how much x growth. Okay, this is the line of your NCERT. Please make a note and keep. I keep telling please, but you people will not make notes. Okay, now this green revolution was given by two people. Worldwide it was given, okay, worldwide by who? Norman E. Borlaug. Okay, he was the person who gave it, and in India, okay, it was given by M.S. Swaminathan. M.S. Swaminathan. M.S. Swaminathan. These were the two people who came up with this green revolution. Yes, father. Very good. Okay. Now, what is going to happen, students? Green revolution had a 3x growth. Green revolution had what? 3x growth. Correct? Yes. Okay. Now, this was mainly done why? Because population was increasing, but resources were not increasing. But what was this green revolution, ma'am? What exactly happened? So, students, green revolution was done in a way that we can improve the growth of our crops. For that, what we should do? First of all, we should give agrochemicals. Agrochemicals. Now, what is agrochemicals? That means chemicals. Okay, thank you, Madhumita. Okay, so students, here what is going to happen, students? Agrochemicals were given. That is, we increase the use of fertilizers, okay, pesticides, so the crops were less damaged. Basically, that's okay, by giving fertilizers, but again, not in a way that it was very highly chemical. Okay, they were all agro-based ones. Okay, this was the thing that they did. After, for a longer time, everyone was very happy. In the 90s, they were like, yes, this is the thing, green revolution. Why? Because as population is increasing, resources also is increasing because of green revolution. We are using agrochemicals. We are using seeds which will give us high yielding. Okay, seeds which are high yielding. This kept continuing for some time. But students, everything has a drawbacks. Do you think, as I told you, do you think, okay, Varu, Praju, just leave the class and rejoin, okay, and don't disturb the class. If you're not able to hear, you leave the class, dear, and then you join and see if you are able to hear. Okay, so now what is going to happen? Other students are able to hear, so you should check your, Bluetooth or whatever you're using. Now, students, all this sounded very nice. Yes, green revolution, everything sounded very nice, but there was a drawback. What was the drawback? The drawback was that since this was continuously happening, but we were not able to keep up to the population. That means population kept increasing drastically, but resources were still not sufficient. Resources were still not sufficient. Clear to all of you? Green revolution is clear? Green revolution was a way where we could grow in a 3x pattern. Now, they did it in a thought process that, okay, as the population increases, let's try to make the resources also in a good way. Okay, resources also in a good way. But what happened? As the population increased, resources was not sufficient. That was the first drawback. Second thing, students, we were using what? We were using continuously agrochemicals. Yes, ma'am. These agrochemicals were very expensive for many farmers to afford. Okay, it was very expensive that many farmers could not afford. So the problem with it is the ones which are, highly chemical, okay, high chemicals are there and they're very, very harmful. Those are cheaper. Okay, those are cheaper. Wow, we achieved the target. Very good. Those are cheaper. But the agrochemical ones, those kind of ones were expensive. Because of that, will the farmer be every time able to go and buy it? No. So, since the agrochemicals were expensive, this also became a problem. Later, what happened, students? With the same variety, we were not able to cultivate maximum because the you know that you understand, no, that the soil also will lose the fertility. Same variety, we cannot keep changing things. So these were some of the biggest problems of green revolution, because of which we had to come to biotechnology, where we had to use gene subtraction, gene addition, gene therapy. Done. Okay, done in the 1956. Okay. Yes, any doubts? Yes, with the existing varieties, we were not able to do anything more. Okay, I will just write that. With the existing varieties, we can't continue the conventional . So this is how we slowly came to our biotechnology. Even though green revolution was accepted and loved by everyone during those time, it was still not completely possible to, you know, come to a complete success. Why? Because even though 3x growth was there, even though agrochemicals were there which were less, you know, harmful, even high yielding seeds we used, and we also used conventional methods, still it was not coming to a rate of how much population was increasing. Okay, yes, if you understood that, shall we start? Okay, very good. Now students, first thing is genetically modified organisms and our crops. Okay, so GMC or your GMO, this was the first thing that was, as I told you, since green revolution came to an end, we had to start with genetically modified crops or genetically modified organisms. Okay, now there were some specialities, just give me a minute. Yes, okay, so this genetically modified crops or genetically modified organism, the first thing was, students, they were pest resistant. Okay, pest resistant. This was the first thing useful of this, because we are doing desired characters. Next thing, they were able to withstand. Okay, they were able to withstand abiotic stress. Okay, abiotic stress. And also their nutritional value was more. Nutritional value was more, and post harvest loss was less. Post harvest harvest loss, all of you know, no, if you are into farming, you know that after, okay, harvesting, there will be a lot of things that will go as, you know, loss because of insect, because of pests. This will happen. But what happened because of this? Okay, because of this, we have to reduce, okay, post harvest loss. So this was allowing post harvest loss. Okay, decrease in post harvest loss. These were the main characteristics. Okay, yes, GMO, when you think of GMC, right? Yes. Yeah, from your username itself you can understand the problem, dear. Okay, problem is clear. Yes, definitely Ramesh, there is a drawback. Everything has a drawback, understand, everything has a drawback, because anything that we extensively use, there is going to be drawback, right, there is going to be drawback. So GMC, GMO was invented because green revolution failed, so they made pest resistant. They were able to do crops which were able to withstand abiotic stress. They did the crops which were nutritionally valuable, and also post harvest loss was less. Also we could increase the mineral content. Okay, that means the quality of the food that we get, we could increase, and we could modify whatever we needed. Okay, modify whatever was needed, right? Yes, less minerals, exactly. So this also we could do. So all these were the changes from biotechnology. Then students, slowly we came into understanding biotechnology in more detail. That is where we could insert and subtract genes. That is, we did gene addition. Okay, we did gene addition, we did gene subtraction, we were able to do, you know, modification of gene, and then later gene therapy, etc, etc. This kind of things we were possibly able to do, right? Yes, okay, possibly able to do. Now let's go forward. Very, very important 6 points I'm going to teach you, okay? Plants, bacteria, fungi, animals whose genes have been altered by manipulation are called as genetically modified organisms. That means we have altered or manipulated their genes, okay? Altered or manipulated our genes. Got it? Yes. Thank you, Bruce Lee, Ramesh. Yes, we will come to that, okay? Ma'am, no emotional damage. It's already done. Okay, genetical modification, that means made crops more resistant to abiotic stress. That's what I told you. I already wrote the points. Reduced reliance on chemical pesticide. We were able to reduce the use of chemical pesticide because if the crop is able to withstand the stress, then it is easier for us. We don't have to all this time spray the chemical, all this thing. Okay? Yes. Okay, yes. Now, help to reduce post-harvest loss. This is what I told you. Post-harvest loss was reduced. Increased efficiency of mineral usage, okay, by plants. This prevents, it does not lead to the soil to be completely useless. That means infertile. Why? Because what happens in the soil, there is lot of micro, macro molecules. Now, the plant, as soon as I'm growing a plant, the plant will try to utilize everything possible for its own growth. Now, on a longer run, the soil will become exhausted. The soil will go to exhaustion. So, because the soil will go to exhaustion, to avoid that, we can improve the quality, okay? That is, increase the efficiency of mineral usage, enhance the nutritional value of food. Example, golden rice, that is vitamin A enriched rice. Any doubts in this, students? So look here, this is what we are going to study, okay? Daffodils are there, genes are taken, plasmids are added into that, we will add it into the rice and we will ultimately get the golden rice, okay? Now I will teach you that. Okay, ready? Ready, get set, go. Yes, definitely. Students, link will be updated, okay? I forgot to check the link. Don't worry, I will share the link with you on Telegram, or I will update the link and share the link again with you, okay? We forgot about the link. Yes. Okay, now gene addition, the first thing we are going to learn is about golden rice, again a genetically modified crop. What is this entire thing? Students, normally we eat rice, we all eat rice, we all love rice, we all like biryani, varieties of rice, but it was noticed that the rice, normal rice that we eat, it is not efficient enough to protect a person who is having color blindness or a person who has vitamin A deficiency. So to this notice, what they did, they were like, okay, let's make a genetically modified crop. That is with the help of rice. That is what they exactly did. To rice, they extracted, okay, beta carotene, which is a precursor of vitamin A. Now, from where did they get the beta carotene? Did they go and buy it from market or Amazon? Of course not. They extracted it from daffodils. Okay, a plant called daffodils which has around yellow color flowers. They extracted it from daffodils. Okay, sweetie, are you here to inform about other channel classes timings? Then you can please leave the class. Okay, you don't have to inform other people about other class timings. You're here, do this job, okay? Yes. I don't like this kind of unnecessary crossing over, because all of you, I know all of you are very smart. So you people will do all this, but don't do it in my class, okay? Avoid such things. Chapter Biotechnology Principles in the chat box. Let me see who all knows the answer for it. Yes. Okay, thank you. Now, that is going to be your agrobacterium tumefacium, because we have to take it into a plant cell, right? So, tumefacients, we are going to take the vector agrobacterium tumefacium, right? Yes, ma'am, definitely. We are going to take a vector and we are going to insert our desired gene. Now, ultimately, what I am going to get after I get this, students, I am going to get my golden rice. Okay, I am going to get golden rice. Now, you tell me golden rice is what? Ma'am, golden rice is enriched with vitamin A. Clear to all of you? enriched with vitamin A. So I'm writing it here only. This is extracted or derived, you can tell. It's not directly extracted, but still these people have a job, okay? Daffodil plant. See, only one word mentioning is there, but still you have to study the whole thing. Okay, you have to study the whole thing. Yes, very good. Okay, did you all like the video? Those who haven't liked the video, quickly like the video. Okay, I will feel very happy that you people are liking and enjoying the class. So students, what exactly happened? Once again, let's go very fast. Just, you know, refreshment. Okay, so rice is there. To that we added beta carotene, which is a precursor of vitamin A, because people who have color blindness or cirrhosis, for these people we can give this golden rice. Instead of taking supplements, we can just do a GMC, that is genetically modified crops. So we took rice, we added beta carotene, which is a precursor of vitamin. Now from where did we get the beta carotene? We took it from daffodil plant. Okay, and that with the help of a vector, that is Agrobacterium tumefacium, we inserted it into the rice and we ultimately got golden rice. Any doubts in this? Okay, any doubts in this? Tell me in the chats. No doubts? All of you? Are you in batch? No. Which batch? Okay. Okay, this could protect a lot of people from issues like, okay, I'll put it in a box. Okay. Yes, they can protect from certain issues like color blindness, any deficiency issues. Okay. This is the whole thing. Okay. Yes. Okay, done. Okay, fine, done. Now, gene addition, we have one more example, that is your beta thuringiensis. But before that, I want to teach you people one more. Okay. Okay, we will do about hirudinaria. After that, we'll go to BT cotton. Okay. Students, golden rice, any doubt? Very simple, no? Beta carotene extracted from daffodils, that's all. Okay, daffodils, that's all. Okay, anyone wants explanation again? Okay, look here. Dear normal rice we take, okay, they have complex carbohydrates. Now to this rice, I'm adding beta carotene. This is a precursor of vitamin A. That means once it enters into my body, it can form vitamin A. Okay, and it is extracted from daffodil plant with the help of a vector that you studied in your principles of inheritance. What is the, who can be a vector, how can they be a vector, how can they get into the cell, all this you studied. Okay, with the help of Agrobacterium tumefacium, what is going to happen? Okay, what is going to happen? Golden rice is going to happen, which is enriched with vitamin A. This will give, if that person has any deficiency issue, that deficiency issue will be cured. Now students, one more extraction I have seen, okay, that is hirudin. What is hirudin? It's an anticoagulant peptide. Okay, it's an anticoagulant peptide. This hirudin is naturally present. Okay, hirudin is naturally present in hirudinaria. Tell me, hirudinaria is in which phylum? And what is hirudinaria? I'll give you a clue. No parking here. Okay. No parking here. Here is that. Yes, very good. Kubera, very good. Yes, hirudinaria. It is naturally present in hirudinaria's salivary gland. Okay, hirudinaria's salivary gland. But we want to synthesize it in a larger amount. We want to make it commercially, or if we want to, you know, sell it in the future also. We cannot take it from leech, right? I told the answer, but yeah. Okay. Yes, all this is in your portion. Don't worry, questions have come in the previous year. That's why I'm discussing. Yes, very good. It is from phylum Annelida, right? Phylum Annelida. Okay, phylum Annelida. So students, what is happening? This hirudinaria salivary gland has this hirudin. But if I want to make it in a larger scale, can I depend on this leech? I cannot depend on this leech. So we can do gene addition here also. That is, I take hirudin gene, okay, synthesize. Okay, this is synthesized version. This is synthesized version because you know that I cannot depend on this, right? Synthesized version I'm taking, and this synthesized version of the hirudin, I will add it to a plant, okay? Brassica napus, which is also called as rapeseed, okay, rapeseed or canola also we can call this. To this I will add, okay, to Brassica, yes, okay. Yes, is a leech. Okay, this one, hirudin area. We haven't reached BT cotton. Please don't go to BT cotton. So students, to this I added, okay, hirudin gene, I have added it to Brassica napus, which is also called as rapeseed, or this canola also we can call it. Now, this, okay, this whole thing is going to happen, and finally the plant is going to grow. I will finally get seed. Yes, ma'am, you will ultimately get the seed. In that seed, student, from the seed, by chemical or enzymatic methods, okay, by chemical or enzymatic method, I will be able to extract hirudin. I'll be able to extract hirudin. Clear? Clear? I'll be able to extract hirudin. Yes, very good. Very good. Okay, clear? Simple thing, nothing much, students. Hirudin gene is there, which I took synthesized gene. I did not take it from this. I took it synthesized, okay? Synthesized gene I added to Brassica napus, which is rapeseed or canola. Now, that will grow. Ultimately, the plant will grow. Yes, ma'am, ultimately, the plant will grow. Okay? Plant will grow. I will get seeds out of it. Yes, ma'am, you will get seeds out of it. Now, from the seeds, I will chemically or enzymatically extract this hirudin and I will commercially sell it. Okay, I will use it for the commercial purpose because I want it in a larger scale because they are anticoagulant. Okay, anticoagulant. Any doubt in this? Also, students, understand this hirudin plays a key role. Okay, plays a key role in blood coagulation. No, so anticoagulant means it will stop the thrombin. Okay, thrombin from that cascade of reaction. That is how we can do anticoagulation. Otherwise, we can't do, no, because it's a cascade, one after the other, one after the other, one after the other. Okay, so this will do the job of inhibiting the thrombin. Any doubts in this? Please let me know in the chats. See, you might not read it from NCERT. That's why I'm asking you. If you have any doubts, please tell me, because questions have come in the previous year and almost everywhere in coaching centers, offline, online, everyone teaches about this. So I'm just adding this. Okay, so what they will do? They will inhibit. Okay, inhibit thrombin. So when they inhibit thrombin, you can understand coagulation will not happen because this is a cascade of reaction. If one step is not there, next step will not happen. Cascade of reaction. Okay, that is what hirudin's job is. Done? Okay, very good. Yes, moving forward. Now, main important thing that all of you waiting for, Bacillus thuringiensis or your BT cotton. So students, cotton, problematic. Okay, cotton is always in problem. Okay, since cotton is very soft by nature, this cotton is always in problem and this cotton is affected. Okay, cotton is affected by bacteria most of the time. Almost all the pests will keep affecting this. Okay, yes, affecting this. Okay, Mrs. Did I tell you? Shumma tell off. Where did all of them go? Yes, okay. So students, bollworm, okay, corn borer, all these people keep affecting this cotton. So this cotton is in big problem too. So what happens, no? This pest also affects, okay, sorry, this pest, I told by mistake bacteria somewhere, okay, pest resistant. So then this pest also affects a bacteria. The bacteria's name is Bacillus thuringiensis. Okay, thuringiensis. Did I make the spelling wrong? Okay, thuringiensis. Now this bacteria, cotton, you imagine, cotton is so, okay, so big. Now what is going to happen? Okay, what is going to happen? This bacteria is so chalak. So chalak means what? So smart. Bacillus thuringiensis, that is when the pest affects, this bacteria will make something called as the Bt toxin. What it will make? Bt toxin that will make sure to do perforations, or you can tell sporulation. That means, students, pores entirely pores. So the pest, obviously, what is going to happen? The pores are going to be made. So whatever outside material is there, it will enter inside the body and the cell will blast open. Imagine a bacteria does something for self-defense. Okay, self-defense became a revolutionary thing in the case of biotechnology. Yes or no? Is this clear to all of you? Very simple thing, no? Yes. So they noticed that, okay, before I write Bt toxin, they noticed that this bacteria, to protect for self-defense, makes crystallize, okay, crystal proteins that also helps in perforation. Okay, perforation, that means making pores around the pest. Now what will happen around the cell? So what will happen? The outside material will enter inside and burst open the cell. Obviously it is not going to work after that, it's going to die. So here they notice that this lead to the formation of Bt toxin. So we can simply use this Bt toxin to protect this cotton worm. Sorry, the cotton worm I'm telling. Okay, cotton. Yes or no? So this was the Bacillus thuringiensis, or your Bt cotton. So how many of you understood this? Tell me in the chat box, Bt cotton. Okay. Now, which are the insects that these people, affect? Okay, they are capable of killing. But students, please understand, this Bt toxin is not harmful for humans because it needs an alkaline pH. I will teach you that. Okay, alkaline pH. Okay, now understand, it will affect most of the insects. No, which are the insects? Like beetles. Okay, lepidopterans, diphterians, all these people, they will affect. Okay, corn borers, bollworms, all these people will affect, but not us. Okay, not us. Yes, clear? Clear or not? Perfect. Very good. Very, very good. Now, look here, students. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as your lepidopterans, tobacco budworm, your armyworm, okay, coleopterans, beetles, diphterians. Now, Bacillus thuringiensis forms protein crystals, that is what I told you, crystalline proteins, during a particular phase of their growth as a part of self-defense. Okay, as a part of their self-defense, and these crystals contain a toxin called as insecticidal protein. Why does this toxin does not kill Bacillus? Because it is in an inactive form. It will only become active when once it enters into the pest. Now imagine, let's call one person as a pest. So the pest has come. Now this will not kill Bacillus itself because it's a toxin, is because this is in a pro-toxin state. That means a pro-hormone. We can tell pro-protein, no? Like how we tell pro-toxin state. That means it will not. It is in an inactivated state. It will only get activated when it is exposed to an alkaline medium. Okay. So why does this toxin not kill Bacillus? Because the Bt toxin protein exists as an inactive protoxin, but once an insect ingests it, okay, it will get activated. That is, inactive toxin will get converted to active toxin. Why it is not harmful for humans? Also, they can ask you a question, because we have acidic protein. Okay, acidic pH in our gut, but here they have alkaline pH. Any doubts in this? What do they do, students? They will make holes. Okay, they will make holes in their entire cells and they will create pores leading to swelling and lysis and eventually death of the insect. Any doubt in this? Death of the insect. Okay, I will write this again for you people as a flow chart. Here I will write it. Okay, now there are some specific proteins which always keeps asking. Okay, it's a PYQ. Remember, what is that? first one, understand Bt toxins are cry proteins, CRY you can write because they're crystallized proteins, no? Now here you have to remember which is for what. That is, see here, there are a number of them. For example, protein encoded by genes cry1AC, cry2AB controls cotton ball worm. Okay, cotton ball worm. Cry1AC. Okay, A for apple, B for ball. So here also A for AC you forget, B for ball you remember, ball worm. So AB, cry2AB. Okay, that is cry1AB is for corn borer. Okay, cry1AB is for corn borer. Correct? Yes or no? Students, you are asking so many things to each other or to me, I can't understand. Okay, so I will write the flowchart for you people. So what exactly is going to happen, students? Let's say the cotton, okay, is affected by a pest. Okay, it's affected by a pest. Now what will happen? This one will go. What we are going to do, we are going to take this Bt toxin, mix it in water and just spread it. Okay, just spread it all around the cotton. Now what will happen? This pest has come to eat the cotton. Okay, this pest has come to feed on the cotton. So when it feeds on the cotton, what is that? In this we know that we have inactivated, okay, inactive protoxin. And from where did this protoxin come? From Bacillus thuringiensis bacteria, which is doing this protoxin as a part of their self-defense. Now this protoxin will enter into the gut, okay, gut of the insect. In the gut of the insect, what will be there? Alkaline pH will be there. In the alkaline pH, this protoxin, okay, alkaline pH, this protoxin will become active toxin. Okay, active toxin. And this active toxin will start making perforations. Okay, perforations means, or holes only you can write. Okay, or pores you can write. Pores in the gut causing swelling and lysis, ultimately leading to death. Okay, ultimately leading to death. Any doubts in this? Okay, this is what happens. Okay, this is what happens. So this is the insect. Okay, this is the villain. Okay, villain, drawing in red color, villain is here. So this villain will come. Okay, this is the problematic fellow. Now we will get inactive toxin from none other than our Bacillus. Okay, Bacillus. Okay, thuringiensis, inactive protoxin will get. Okay, got it? Clear? Yes, done. Done. Yes, very good, very good. Okay, this is how it will look, students. Can you see normal cotton, infected one, because here the pest has infected. Okay, and this will lead to complete drying of the cotton, and it was a big problem for the farmers back then. They did not know what to do and where to fix it, so they were in lot of tension, and that's when finally Bt toxin has come. This will not harm us. We use Bt toxin for brinjal, growing of brinjal, okay, even growing for tomato. These things we use it, but it's not harmful for us because we have an acidic pH. Please remember that. They could be asked as a PYQ question. That time don't be like, huh, why? So don't ask why to each other. Instead answer the question. Okay, yes, just give me a minute. Okay, that's fine. Okay, perfect. Now we'll go to one complicated topic. Okay, little complicated topic in this chapter. Other than that, everything is baby, students, baby topics. Okay, that is your gene suppression. So what we did now, we did golden rice, we did hirudin, we did Bt cotton. All of this was gene addition, right? We were taking something from someone and we are doing something. But now we are going to do gene suppression, telling that this gene is problematic. I don't want this gene, so I will suppress this gene, or I will stop the RNA. Okay, I will do RNA silencing so that translation does not happen. Because if translation happen, I cannot control anything. So to avoid that entire step, we are going to do something called as RNA silencing. That is going to be done by your Melidogyne incognita. Okay, Now, students, this Melidogyne incognita was a big problem for tobacco plant. Which plant? Tobacco plant. So, all the tobacco plants' roots, okay, all the tobacco plants, root systems. Okay. Let's say this is the root. Okay, all the roots of the tobacco plant started seeing knots, like this knots on the root. Now you might be thinking, ma'am, what is there if there is one knot? But students, understand, root has to do the absorption, root has to do, okay, continuous, transport of all the substance to the top, right? So if the root have knot, then how is it going to do that? So they noticed that this root is starting to get root knot disease, okay? What disease? Root knot disease. And this disease was caused by who? None other than your Melidogyne incognita. Okay, Melidogyne incognita. This root disease was caused by a nematode. That was the nematode. Now it has become a big problem, students. All of you know the use of tobacco, and it is also one revenue source for us, right? Because the government will get some money from all this selling of all these things, no? So it was big problem then, back then, that all the tobacco plants, all the roots started getting this knot disease, okay? This nematode started entering into the plant, yes, entering into this tobacco plant. Now people started thinking, what can we do? But we realized that we cannot do gene addition, we cannot do so, we have to do something called as RNA interference or RNA silencing, okay? What we did, the only solution was RNA interference, okay? RNA interference, that is, we have to basically silence the RNA so that it does not do translation, okay? So that was done with the help of RNAi, we call it RNAi, that is RNA silencing. This was the only method, which is also known as your gene suppression. Okay, yes, very good. RNA interference or gene suppression. Now, what is this RNA interference? Where did it all come from? How did people even think of it? Understand, students, in all eukaryotes, including us, okay, in all eukaryotes, this happens as a natural cellular defense. It is a natural process that happens in our body because we do gene regulation. We decide when the gene have to be switched on, switched off. All this we do, no? But in eukaryotes, this happened in a more major stage. That is, during the translation itself, some of the RNAs can completely silence. All this can happen in our body. So all eukaryotes, normally these things happen as a part of the cellular defense. Okay, is this clear to all of you? From where did this whole topic come? From all the eukaryotes. Okay, all eukaryotes. Okay, in all eukaryotes, this is going to happen as a part of cellular defense. Okay, cellular defense and gene regulation. Gene regulation. Clear? Perfect? Yes, very good. Very good. Okay? Very good. Now, students, this gene regulation or this RNA splicing is a more complex process. I know many of you might have studied here and there 1 or 2 lines, but let me tell you, if you go in depth to this is a very big process, okay? Because there are some regulatory RNAs like your siRNA, microRNA. In that, your siRNA is taken from the long nucleotide. That siRNA will go and join with an agronut protein that will entirely form RISC. So this is a more complex process, okay? But I will try to make it easier for you. But again, lot of things I have to skip to make it easier for you people, okay? But don't worry, nothing will get skipped from your NCERT. But the process will go a little off, okay? Because it's more complex, okay? Now, RNA interference, for this, we need the most important thing that we need is double-stranded RNA. What I need? Double-stranded RNA, very important topic. Question can come from this, okay? I saw a PYQ also on this. That is double-stranded RNA. For this double-stranded RNA, that is the source. Okay, I need a source for this, no? From where will I bring this? Students, most of the time, let me tell you, it can happen inside the cells, but sometimes your entire this process, no, everything happens inside the cell. Sometimes we give it from outside also. The first source is from virus, okay? Virus, this is given in your NCERT, and second one is transposon. Okay, transposons. These are mobile genetic materials. Okay, mobile genetic materials. These two lines are given in your NCERT, so you have to know the source from where I can get my double-stranded RNA. Okay, because double-stranded RNA is not something common, no? So I need to get my double-stranded RNA from somewhere. Okay, double-stranded RNA from somewhere, I need to make sure to get it. Now, listen carefully. There are two types. Okay, there are two types of this regulatory mRNA. Okay, regulatory mRNA. One is siRNA, okay, and one is microRNA. MicroRNA. Both of these people at one stage of their life will meet a dicer. Whom they will meet? Not dinosaur, dicer. So this dicer is an endoribonuclease. Okay, endoribonuclease. This dicer will do the job of cutting. Okay, it will cut the mRNAs. Okay, this siRNAs, no, they will cut the RNAs. And after they cut, two strands will be there, no? I'm just telling the general, then I'll take you again to the steps. Now, two strands are there, not because it's a double-stranded RNA. It will cut the strands. It will up to be 21 nucleotide. Okay, it will cut the strand. After cutting the strand, one strand will be taken. Obviously, no. Out of two strands, one will be selected. This one strand can be called as the guide. Okay, guide strand. Now, this strand will be taken up by your, I told you, no, by your agronut protein, which you don't get, which you don't have to remember. That protein will come and take this up. Okay, take this up. And students, this combination, okay, this combination with this guide RNA with some other proteins together forms the RISC. Together forms the RISC. Tell me the full form of RISC. I will give another simple explanation. Don't worry, just understand that these terminologies you might come across later. So I don't want you to be at risk. Okay, tell me. Yes, what is the full form of RISC? I've taught you this before also. RNA induced. Yes, rest you can tell me. RNA induced silencing complex, right? RNA induced silencing complex. Clear? Students, what are we basically trying to do? We are making sure that these people do not go to the next step of translation, and we make sure that they stop. They silence it. Okay, silence it. That means we will just stop this whole process. This happens in our body on a regular basis. It's a natural process. But since this Melidogyne incognita is affecting the tobacco worm, we have to do this process for Melidogyne incognita. Okay, now students, look here. Look here. This is your viral RNA. Okay, this is your viral RNA. This is our viral dsRNA, which we already formed and kept. Okay, so now imagine if Melidogyne is going to come. We are already prepared with a double-stranded RNA, which will make sure to not let this RNA go to a translation stage. Okay, translation stage. Clear? Okay. Thank you so much, Abdul. Yes. Now here, what is going to happen, students? Remember, we have already made this we have already made this double-stranded RNA. How did we make this double-stranded RNA? We already took the virus, okay? Virus, or we have taken transposons and we have made it. That means we have made sense strand and antisense strand. Okay, sense and antisense is made. Together we have the dsRNA already ready. Now this fellow will do the job of silencing. Now what is going to happen? Happily, your nematode is going to come. Okay, your nematode is going to come. Nematode is going to come and release their viral RNA. Okay, viral RNA. Now, will this viral RNA go to the next stage? Of course not. Because look here, all of you. Whatever I told, no, just keep it in mind. Simple thing. This is a viral RNA. This is the strand. I told you it will meet a dicer. That means it's an endonuclease which will chop these people off into small strands, okay? That will lead to the formation of siRNA generation, small strands of RNA, you know? So siRNA formation. Then they will go together. I told you many proteins from plus this agronut protein together will form the RISC. Okay, the RISC assembling. Can you see RISC assembling? Now this RISC will go to the RNA targeting and then it will do splicing. Okay, it will slice everything and it will make sure that this viral RNA does not go for translation. That's all, students. Too much if I go also, you will go crazy. Okay. Now this RNA has released, no? To this RNA, who is going to come? This double-stranded RNA is going to come. Wait, I will just draw this little more better. Okay, so you people can understand. Yes. So, okay, this is our DNA. We'll tell the DNA to make RNA. It will not make just one RNA. It will make 2 strands of RNA. The 2 strands of RNA will be bound to each other. And this will lead to the formation of double-stranded RNA. Okay, double-stranded RNA. Okay, double-stranded RNA is already there. This is the main process. Okay, already went crazy. Yes, now just listen to this. It is easier. Okay, DNA is there. DNA will do what? DNA will make RNA according to your central dogma, according to your genetics. Okay, now this dsRNA is formed. This is again sense and antisense is there. Okay, strands are there. Complementary, no? So strands are there. Now, next what is going to happen? This nematode is going to come. Okay, this nematode is going to come and nematode is going to release its viral RNA into the tobacco plant. Okay, nematode will come, it will release its viral RNA. So this viral RNA will not be able to go forward because this dsRNA will attack this. Okay, dsRNA will attack this, will do chopping, cutting. Okay, will do slicing. Okay, it will do slicing and it will not. Can you see that slicing is done? It will not let this people continue with translation. So no translation, that means no disease. This much can you remember? This much can you remember? Yes? Yes or no? Students, I told you this is a little complex. The main problem is teachers cannot explain the whole thing because it becomes more complex. Teachers cannot explain half because you will not understand. So the concept is given in NCERT in a very differentiated manner, but just try to understand at least RISC, at least know the full form of RISC. Okay, this is an whole complex. RISC is there, dicer is going to be there, dicer is going to do the cutting of here. Okay, siRNA. If you want, I'll share a video link to all of you on Telegram channel. It's an animation video. See if you can understand. Okay, see if you can understand. Yes, it will destroy. Okay, it will destroy the viral RNA. Okay, it will destroy the viral RNA. Basically, understand, we have to destroy this viral RNA. For that, we are going to take this dsRNA. This dsRNA will be cut into small pieces which will form the siRNA. To this siRNA, one protein will come and attach. It will take only one strand and go, telling that I selected you. This 2 combination is called the RISC. This RISC will go further. What it will do, with the help of many other things, it is going to go and join with this viral RNA. Understood? To this viral RNA, if RISC comes and joins, then it cannot continue because this RISC is going to do the splicing, like slicing of it, and it will not further continue. Okay, not further continue. Clear or not? Clear? DNA is there, it will form RNA. Once again, I'm explaining, listen carefully. Okay? Okay, listen carefully. Not splicing, slicing. By mistake, I told splicing because I taught genetics yesterday. Slicing it will do. Pieces. Okay? To make it into multiple pieces. Okay, sorry for that. Not splicing, slicing. Listen to me carefully. DNA is there. DNA will form RNA, but we wanted double-stranded RNA. For that, we have taken 2 sources. Neither we can take an RNA virus or we can take our transposons. Let's say we have taken transposons and we form the double-stranded RNA. This double-stranded RNA is capable of making sure that the RNA silencing happen, or I interfere with the RNA. Now, nematode is going to come. It is going to release their viral RNA. When this viral RNA is going to be released, what happened? A RISC complex is formed. Okay, how this RISC complex is formed? With your siRNA, I told you siRNA will be cut with the help of dicer. Okay, dicer. Dicer will cut this siRNA and this siRNA will be attached with another protein. They will form a complex called as RISC complex. Okay, RISC complex. This RISC complex will go and join with your viral RNA and it will do slicing. Okay, it will make it into multiple pieces so that translation does not happen, because if translation has to happen, RNA has to be transported from the nucleus to the cytoplasm and then translation will happen there. If you give pieces, what will it do? Okay, if you give pieces, how will it do the translation? So translation will stop. That means no protein, no disease. Clear? Simple way I have told. More than this simple I can't tell students. This is the simplest way possible. Okay, simplest way possible. So here also you can see viral RNA, viral dsRNA, and then how it is meeting a dicer, how it is making small pieces. That small pieces will be attached with the help of a protein. That protein, that entire complex, only one strand will be removed. Can you see? Out of these two strands, only one is taken. Why? That is called the guide strand. We will select only one strand. Okay? Got it? So that one strand will go and attach to the RNA, so okay, which we are targeting, and then we will do completely slicing. Simple as that. Now I am targeting you. I will come there. Before that, me and my friends already made a complex. Now we all will come. We have targeted you. So we will separate into two gangs. Okay, I am the guide because I know you. Okay, I will become the guide strand. The other fellow will go somewhere and I will come with my friends. I am the risk complex. Okay, I am coming towards you. I am targeting you. I will join with you and I will break you into emotional damage. I will do. I will do slicing. Now, can you go forward and do something? You cannot do something. Okay, this is the simplest example possible to tell. Okay, this is the Melidogyne incognita. So students, now NCERT if you read, you will understand that, okay ma'am, what you told is better. So look here, several nematodes parasites a wide variety of plants and animals including human beings. A nematode called Melidogyne incognita infects the roots of tobacco plant and causes what? Great reduction in the yield. Obviously, if root is not healthy, the plant also is not going to be healthy. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, which is called as RNAi. RNA interference takes place in all eukaryotic organisms. Very important question. All eukaryotic organisms as a method of cellular defense. Okay, I have just written NCERT lines for you. Okay. Okay, now this method involves silencing of a specific RNA, okay, silencing of a specific mRNA due to the complementary dsRNA, okay, double-stranded RNA molecule that binds to and prevents the translation of the mRNA. Easy or not? Now, are you able to understand? What they are going to do? They are going to bind with the mRNA and tell that I will not let you for translation. Simple as that. Easy. Please put thumbs up in the chat box. The source of this complementary RNA could be an infection by a virus. Okay, that's why I wrote RNA virus. Okay, infecting a virus, having RNA genomes, or a mobile genetic element, that is a transposon. Okay, transposon. So I will write here. Okay, I'll write here. Mobile genetic material I have written. Okay, here a virus infecting RNA cell which has RNA genome. Okay, these are the two sources of getting your dsRNA. Fine students, peaceful, easy, easy. Okay, very nice, very nice. More thumbs up came. Now, biotechnology and its application in medicine field. Okay, how these people are going to help. Before that, let's drink some water. Okay, let's drink some water. Okay, yes. So, gene suppression can also be given. One more kutti example they have given. Flavor. Okay, flavor. Savor tomato. Okay, tomato. I have not seen questions on this, that's why. So flavor savor tomatoes, you know, usually if you keep tomatoes, after some time immediately it will start becoming very soft and mushy. It will be good for you making a soup. You cannot make like chop and make chicken curry or fish curry. Fish curry, I don't know if you put tomatoes. Okay, chicken curry or like paneer. You cannot make butter paneer with this tomato because it will be so mushy. So to make sure that this tomatoes does not get soft very fast, we can do gene suppression. Okay, so that also is a process, but I have not seen any of the places. Before I used to teach, but now I have not seen questions on that. But just remember, flavor savor tomato. Okay, yes. How does this look like a sister chromatids for you people? Huh? You people are too much into biology, I feel now. Okay. Yes. Fine. Okay. Shall we continue? So that also just keep in mind. Okay. Now students, medicines. Understand biotechnology when we talk about, we understand that okay, there are a lot of benefits. Okay. Same thing happens in medicine field also, students. That is, the medicines that we get, no, they are mostly the medicines that we get with biotechnology. They do not, our body does not react to it. Okay. React to it. Okay. Immunological response is comparatively less. Okay. Comparatively less. Okay. And these medicines are definitely cost effective. Okay. They are cost effective. And they can treat multiple diseases. All this is there, but you should also understand that the most important thing why we use biotechnology is for large scale production. Okay. Large scale production. Okay, large scale production. These are some of the reasons for using, okay, reasons for using your medicines. Okay, that is biotechnology based medicines. Now students, around 30 medicines, okay, 30 therapeutics. Okay, around 30 of them were found. Okay, out of which 12 is sold in India. 12 is sold in India. Okay, tomorrow if you go and ask some medicine, that pharmacy person, which is the biotechnology medicines, he is not going to know. Okay, yes. Okay, autoradiography we'll come into. Rani. When I teach you diagnosis, I'll teach you autoradiography also. Okay, yes, we'll keep this flow as of now. Yes, so biotechnology, therapeutics, that is your medicinal purpose we have started using. Why? Because they do not give high amount of immunology. That means the body does not respond, okay, like profusely for this medicine. They are good and they are cost effective. They are also produced in large scale production is happening. Okay, 12 is sold in India according to your NCERT. 30 were made, but 12 is sold in India. Now, most important, insulin. Insulin. Now, what do you know about insulin? You know it is in the pancreas. Okay, insulin, okay, insulin is given to a person who has diabetes, okay, diabetes mellitus, not diabetes insipidus. Diabetes insipidus is ADH, okay, diabetes mellitus, that means sugar, okay, sugar we tell, no, normal language we tell sugar, diabetic patient. So diabetes mellitus, we give that person with insulin. Okay, insulin injections we give them. Now, where this insulin comes? Obviously from pancreas. Our body makes it, but sometimes our body, islets of Langerhans. Okay, islets of Langerhans are the set of cells which are going to release your insulin. Okay, now students, sometimes what happen? Obviously the insulin levels body will not be able to make. Because of which, because of the hyperglycemic condition, the glucose content is too much high in the body. What will happen? The body will not able to break down all this glucose and store it as energy and all of that. So we have to give insulin from outside. So people are like, let's make genetically modified insulin, because high commercial value is there. You know, all of them. Now it has become like a status that, oh, I have diabetics. It's become like, you know, how I have hair. It has become so common among people about, like, you know, in people who are at the adolescence, puberty age, at adult, the old age, all of this people are carrying it like a, you know, like there's something to be proud about. Okay, there's something to be proud about types, right? So same thing here also. Insulin is highly commercial value is there. So we cannot make, okay, we cannot make, you know, things like, okay, 1 or 2 is there, so it's fine. No. So we have to make it in a larger scale. So for that, what they did genetically modified insulin, students, but it was a task to make it. Why? Because this insulin structure is different. That is, they have an A chain. Okay, they are, first of all, understand insulin is a peptide hormone. Okay, it's a peptide. This is the reason why you do not take insulin orally. Not taken orally. Why? Because your stomach will digest it. Okay. Your pepsinogens will be active. Oh yes, something is coming, peptide, I need to break down. Okay, break down. So here what is going to happen? Peptide it is, so it is not taken orally because it will be digested in our body. So students, A chain is there, B chain is there. Okay, A chain and B chain is there. So this is how our insulin is there, and this both A chain and B chain are connected with disulfide bonds. Okay, disulfide bonds. Disulfide bonds. But this A chain and B chain is also connected with the help of a C chain. With help of what? A C chain. This is the structure of insulin. Okay. Yes, I have not had my dinner, so my insulin would have got confused what is happening. Okay, so this is how your insulin looks like, students. Now many people tried, okay, we have to make insulin, what to do. The biggest problem that they were facing is the C chain. Okay, the biggest problem they were facing was because of the C chain. Okay, the C chain they were not able to make, but understand, later we realized that this whole structure is at a pro stage. That means in an inactive form. No, inactive stage it was. Okay, students, if I take break also, there's no use, so we will continue the class and finish it off. Okay, because I have to go home only and eat. I have to go home only and eat. Okay, yes, clear. Okay, what is the biggest problem that these people faced? These people faced problem was that this C chain was not at all, they were not able to process this C chain. That was the biggest problem. But they noticed that this entire structure is in an inactive stage. Okay, inactive stage, or we can say it is a pro-hormone stage. Okay, pro-hormone stage. Since we are genetically making insulin, then there came a company called as Eli Lilly. Who? Eli Lilly. What is the year? 19. Let's see. You take a break. I can't take a break. Tell me. Which is the year? This year has been asked multiple times. Please make a note. 1983. Okay. So Eli Lilly was a company, not one girl. Okay. Don't write, oh, Eli Lilly, a girl who made this. Eli Lilly, yes, difficulty in assembling. Very good, Kubera. So they had a problem, as I told you, the C chain, the entire assembling was a difficult task. Because of this, Eli Lilly company realized one thing, that, look at it, this is in an inactive pro-hormone stage. Then why do we need C chain? If A and B chain can do the process, why do I need C chain? So Eli Lilly was very smart. They worked on E. coli. Okay, they worked on which bacteria? E. coli. To make humulin, okay, humulin that had only A chain and B chain. So what did they take, students? They took, let's say, E. coli, okay, A, I'm just labeling it B, okay, E. coli B. From both of them, from one they got the A chain, okay, they extracted the A chain. From one they extracted the B chain, and finally they brought A and B chain together with a disulfide bond. Okay, this is not like this. Basically, you can draw it as a box only. Okay, they got the A chain and B chain. They got a smaller A chain and they got a bigger B chain. Okay, and they kept it together with the help of disulfide bond. Clear? Easy, no? This is? Yes, ma'am, very easy. Okay, very simple, no? They bonded with a disulfide. They kept it together with a disulfide bond by not taking tension of C chain, because C chain was kept, the C chain was kept during a pro hormone or a pro stage, okay, inactive stage. So during the active stage, these two people are going to help. Then why do I need C chain? So the difficulty, okay, difficulty in assembling was solved by not, okay, but by not synthesizing C chain. Clear to all of you? Easy? Easy? Yes or no? Thank you, Ajay. Okay. Thank you, Bruce Lee. Yes. Okay. Perfect. Thank you so much for your messages. So sweet of all of you. Yes. So students, diabetes mellitus, all of you understand your normal diabetes. For that, we will give insulin. It is given intravenous. That is mostly in injection. Okay. It is not given orally. Please understand. It cannot be given orally because it's a peptide, and peptide means it will get digested in our body. So pancreas, islets of Langerhans, they form insulin. It's peptide not taken orally. So what is the structure of insulin? A small amino acid chain, a bigger amino acid chain, both of them are bonded with a disulfide bond. This disulfide bond, both the A chain and B chain was also connected with the C chain. So most of the people found it very difficult to make the insulin chain, but the initial stages people made the insulin, okay, made the insulin with the help of cattle, okay, cattle. What happened? Okay, here something is missing. Proactive. Okay, yes, students, first they did it with the help of cattle, and it was not at all practical. Okay, it was not at all practical. Why? Because many people were getting allergy because of it. Okay, from cattle they try to extract. Okay, extract from cattle. Okay, extract from cattle, but it was not practical because it was causing allergic reactions. Okay, allergic reactions. Done. Okay, it was causing allergic reaction. That is why we had to make humulin. Okay, humulin, and that the main problem when they were doing this was difficulty in arranging. Okay, the main problem was in difficulty in arranging the C chain. So what they did, the Eli Lilly company, it is a company, not a girl. Eli Lilly company in 1983 solved this by making humulin with E. coli by not even having the C chain, because C chain was present in the proactive stage. Okay, thank you so much. Yes, yes, thank you so much, Tanuja, Ajay. Okay, now gene therapy. Okay, gene therapy. What is gene therapy? Now, before I go to gene therapy, we should know from where does this come. Okay, gene therapy concept. So, students, there was a kid, okay, 4 years old. 4 years old kid. Okay, I forgot the child's name, student. I had read about the child. Okay, now she is, I think, 53 or something. Okay, I have read about this child, but I forgot. Okay, forgot. Yes, okay, very nice. So in this 4-year-old child, okay, 4-year-old child had a problem called as SCID. Okay, SCID. What is this? Severe combined immunodeficiency. Okay, severe combined immunodeficiency. Okay. Now, students, because of this, okay, because of the severe combined immunodeficiency, how did this kid come? First of all of you are not commenting, okay, after class. You people will tell, yes, ma'am, so sweet of you, you are standing for past 4 hours, 5 hours. Last genetic class, we took for 8 hours, okay, without sleep, till 2 o'clock. Both sir was taking class, I was awake in the chat box, and you people didn't even have the little courtesy to comment and let us know. Only some of them commented, and I'm going to give a shout out to them today at the end of this class. Okay, so severe combined immunodeficiency, this came because of ADA deficiency. Because of what? ADA deficiency. That is adenosine, you can tell adenosine deaminase deficiency. Now, what is the use of this? Why did this deficiency cause an immune, okay, deficiency? That you have to know, no? That you understand, students, this adenosine deaminase plays a very crucial role in lymphocytes, that is work with your immune system, okay? It is actually playing a very, very crucial role, okay, in your lymphocytes. Okay, in your lymphocytes maturation, we can say, or lymphocytes differentiation and maturation. In the whole process, okay, these people are very, very helpful. Okay, helpful. But if this is not there, then it is definitely going to sacrifice the immune system, right? That is going to sacrifice the immune system. Yes, ma'am, it is going to sacrifice the immune system. So that was the problem here. Okay, that was the problem here. The kid had SCID. Okay, the kid had SCID, because of which, sorry, okay, yes. So adenosine deaminase deficiency was there, and it is to be correct, it actually removes a toxin, okay, for this lymphocytes to go further. That is what the thing is. Okay, so this ADA deficiency led to the severe combined immunodeficiency. Now students, many people can suggest many things, no? Now they suggested that, okay, let's do one thing, because this is an enzyme, let's do one thing, let's do enzyme. What can we do with enzyme? Tell me in the chat box. Very good. Yes, full energy. Okay, sir is there. Okay, very good. We can do enzyme replacement therapy. Doctors told that, okay, let's do enzyme replacement therapy. But students, do you think it's practical? Imagine in my body there is a natural enzyme. Okay, that I have to go and get injection every time. It was not practical. So enzyme replacement therapy was not practical. But again, I'm telling you different solutions that we could go. Then the second thing was, because lymphocytes were affected, let's go for bone marrow transplantation. Let's go for bone marrow transplantation. This was the next thing told. Okay. Okay, this is also one solution they told. The last thing that they told was gene therapy. Okay, last thing they told was gene therapy. That is, ADA deficiency is there. Okay, let's alter the gene. Let's alter the gene. Let's say in the gene, ADA deficiency is there. Let's modify, add the gene. Okay, and then add it to this 4-year-old kid. 4-year-old kid. Now students, understand, all your lymphocytes, WBC, RBC, everything has a lifespan. So after the lifespan, how is the kid going to survive? Because you inserted this gene to a 4-year-old kid, obviously the body is going to perform normally, but everything has a lifespan. Will it be a permanent solution? No, it will not be a permanent solution. Okay, it will not be permanent. All these are temporary solutions. For a permanent solution, the ultimate thing that they could, okay, they could have done is that for a permanent, okay, for a permanent solution, the only thing that we could have possibly done is do gene therapy. Okay, gene therapy itself. Okay, do gene therapy at an embryonic stage. Okay, at an embryonic stage. Clear? This was a permanent solution. Any doubts in this? Please tell me. The Ajay, you can search that 4 years kid. Okay, actually that kid is there. Okay, that kid is there, and you can see the photo of that child. I forgot the name, yaar. Some, you know, this, for me, this names are a little difficult to remember. Okay, different solutions that they gave was what? First solution they gave were enzyme replacement therapy. But again, all of this are temporary, because the child has to keep coming over and over to do the process, right? Enzyme replacement therapy, bone marrow transplantation, and your gene therapy. Gene therapy is a good solution, because whatever gene is missing, that is your ADA, we added and we inserted it to the child. But the problem is, since everything has a lifespan, there can be possibility, okay? There can be possibility that this will not, after some time again the child will have to go back and tell that, okay, please give the gene again, gene therapy has to be done. But the permanent solution that we can always go for is gene therapy at an embryonic stage. Clear? Okay. Sure, Abhishek, don't worry. Tomorrow some big surprise is coming for you people. Okay, big surprise is coming. Done, dear? Clear? How many likes are we? Oh my god, so less. Why aren't we 300 likes? Make it 300 likes. All of you quickly in the chat box. Quickly in the chat box. Students, I'll get some water. Till that, please be on. Stay here. Don't run away. Ashanti D. Silva is the name. Huh? Okay, I'll just get some water. Please stay here itself. Don't run away. Whoever run away. Yes, good job. Okay, good, good. None of you ran away. Okay, now we'll continue. We have very small portion left, right? Okay. One second, students. Okay, fine, done. Okay. Yes, this is done. Gene therapy is done. Okay, now molecular diagnosis. That is, students, different methods are there, no, to find different diseases. Now when we speak about HIV, I talk about ELISA. When I spoke about typhoid, they told that to confirm the test there is viral test. So there is molecular diagnosis. There is definitely a conventional method. What is your conventional method? That is, let's say if you have a disease, what will the doctor tell you? Please give your blood sample, please give your urine sample, right? This is the normal conventional method. This is the preliminary test, like how you used to do in chemistry, no? First you used to do titration, you used to get the preliminary, not in titration, I guess, salt analysis. You people used to do preliminary test, no, to confirm which salt this is. Okay, we used to do preliminary test. After that, we used to do the reaction. So like that only, this is a preliminary stage. That means, okay, this is the first stage to identify if I have a disease. But in the deeper, if I want to go, I will try different, okay? Yes, HMPV news is coming. 2 cases in Bangalore spotted. One is a 4-month kid and one is an 8-month kid. But both of them had a, that is bronchi issue. So they are not telling that it is a threat like COVID. But then, yes, HMPV is still on threat because we don't know what's happening in China. I saw a lot of new students, but the thing is, some people are telling that it does not spread in China. This is all, this is all fabricated and exaggerated. Some people are telling, no, it is spread in China. So there is lot of, add-ons to this, because people want to create drama. People want to create that, the fear in people, because if you make the fear only, you can sell what you're selling, no? So like that only. So same thing is happening. So don't worry, if something is there, they will tell it, okay? If something is like going, an outbreak is happening, definitely we'll get to know. But half of the things are exaggerated, okay? Exaggerated. That is the thing. Okay. So some of the, okay? Yeah, new virus. Conventional methods. Tell me which are the conventional methods. That means a preliminary test that you will tell. Come on. First thing, what did I tell you? Urine test, blood test, serum test. Right, these are the things that normally people, like as soon as you go to a hospital, now you want to check if you have thyroid, you will give your blood and come. You want to check your diabetics, you will give blood and come. Okay, you want to check if you have kidney stone, you want to check if you have, UTI, you will give your urine test and come. Okay, same way you will give serum test. So these are some of the preliminary tests, but we need to go further and understand in depth what is there. For that, we will do molecular diagnosis. Give me some examples of molecular diagnosis. Now, if you want to check if a person is HIV positive, what will you do? You will do the first technique, which is ELISA. What is the full form of ELISA? Enzyme linked immunosorbent assay. Okay, enzyme linked immunosorbent assay. What is this, students? The principle of enzyme linked immunosorbent assay is an antigen antibody principle, no? Antigen antibody binding. Okay, this is the principle behind this. Okay. This is the principle behind this. Yes, very good. Very, very good. Yes, very good. Next one is, ma'am, obviously PCR. Polymerase chain reaction. Just now my students studied PCR. What is the steps of PCR? I always told you, you call your friends Bay, no? Instead of that, you call Day. Okay, so DAE. That is denaturation. Denaturation. Annealing. And extension. Okay, extension. All this is change of temperature. We are not changing the process, or we are not lifting the tube and seeing every time. No, just change in temperature. Okay. Polymerase chain reaction is where a small portion of a strand also we can make copies. Okay. We can make multiple copies of it, right? Polymerization, complete copies of it. That is your PCR. Any doubts in this? Please tell me in the chat box. Good job. All of you understanding? Yes. So PCR we use mainly for our amplification of our DNA strands. Okay. To make multiple million copies. Okay. This is our second thing. Now students, other than this, we have a probe, which is an autoradiography, which I taught you just 2 days back in our now where do I go for this? Okay, 1 second students. Yes. Okay, I'll write it here only. Autoradiography, I just taught you people in your DNA, right? DNA fingerprinting. What is autoradiography? That is one of the steps of your DNA fingerprinting. Autoradiography. Now students, autoradiography totally works on one concept of probe. Probe. Remember probe, all of you? Probe. Now what is probe? It is basically a complementary strand, right? I told you that there is repetitive sequence. Okay, repetitive sequence. Now let's take 3 examples. Who wants to be my example? Who wants to be the example? Okay, let's take Kubera, Meghna and Rani. Okay, Kubera, Meghna and Rani. This is Kubera. Okay, Kubera. Kubera is in orange, okay? Meghna is in blue and Rani is in pink. Okay, Rani is in pink. Now these 3 people's strands are there. Now students, imagine, let's say a combination, a repetitive sequence is there, A, G, T, T, C, G, C. This sequence, let's say in Kubera, it's repeated 10,000 times. Okay, in Rani, it is repeated only 5 times. Okay, and in Meghna 5 times. In Rani, it is repeated only 500 times. Now, from this, what I am going to do? I am going to make a probe. Okay, I am going to make a probe which is complementary to this. So, A will bind with T, C, A, G, C, G. This is a probe. Okay, this is a probe. Now, what it will do? Let's say the criminal. Okay, who wants to be the thief in this? Okay, let's make Kubera the thief, because the boy only, no? Kubera? Because I can't tell students, your YouTube names are so fancy. Even if you put dinosaurs, it's okay. But if people put some boy name for girl, girl name for boy, very difficult. Okay, so here, let's say, out of these 3 people, let's say Kubera's, okay, sample we got. Okay, some hair strand, something we got. Okay. Okay, Bruce Lee became the thief. But then I have to add 1 more strand. Wait. Don't cry, I'll put your strand. Okay, let's say 5,000 times. Okay, students, imagine from the crime scene, we got Bruce Lee's blood sample, or we got some hair or some sample we got. Now, what I'm going to do? I know that this is repeating 5,000 times in Bruce Lee, okay? So, this will go and correctly attach to the DNA strand where repetition is happening, and then finally I will be able to find in the autoradiography who is the thief. Okay, who is the thief? So Bruce Lee became the thief. Okay, fine. So this probe, students, please understand, this is radioactively labeled. Okay, radioactively, okay, labeled. Clear? Yes or no? So, this radioactive labeling we will be able to see under the autoradiography, and we will be able to understand that, okay, 5,000 times this repetitive sequence is there. It's not necessary that all of these people have the sequence also. Maybe, Rani does not even have the sequence. Rani has a different repetitive sequence, but that 0.1%, you understand, that will keep differing. Okay, that will keep differing. Clear? Yes, this is your molecular diagnosis. Rani, is that clear? You had asked me the doubt. Yes, clear? So this is your autoradiography. Probe is basically complementary to your VNTR probe. I mean VNTR, this one, okay? Variable tandem repeats. Now, presence of pathogen, bacteria, virus is normally suspected only when the pathogen has produced a disease symptom. Now, all of you know, in the case of malaria, 3 to 4 days we get recurring fever. We do not get constantly, continuous fever is not there. Recurring fever will happen whenever hemozoin will be released from the RBCs, we will get recurring fever. Now during that situation, we only understand once we get fever. Now how do I know I have an allergy? Only if I sneeze, like that. Okay. Now by this time the concentration of pathogen is already very high in the body. Even though the symptoms have started, that means they have already started multiplicating. However, very low concentration of the bacteria or virus at the time when the symptom of the disease are not yet visible can be detected by amplification of neither nucleic acid by the PCR or can be detected by your ELISA. Okay. PCR can detect very low amounts of DNA. PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutation in genes in suspected cancer patients. So in two things, very important, very important. What is it? Cancer patient as well as AIDS patient, they are using PCR to detect. That means people who have doubts that okay, something is there, for them AIDS and cancer patient, we are using PCR for amplification to understand if there is any problem. Okay, if there is any problem. Clear? Yes. Now it is a powerful technique to identify many other genetic disorders. A single-stranded DNA or RNA tagged with radioactive molecule, that is a probe, is allowed to hybridize to the complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not be complementary with the mutated gene. Now let's take an example. Students, this is a very cruel example, so I will just take some cartoon name. Okay, let's take Tom and Jerry. Let's take Chota Bheem. Okay, let's take Chota Bheem. So this is Chota Bheem. I cannot draw any of the cartoon, by the way. Okay, Chota Bheem is masculine, no? Very muscle man he is. Okay, Chota Bheem. He ties a punche like this. So yeah, leg. This is Chota Bheem. Now Chota Bheem has a problem. Okay, what is the problem? Let's say Chota Bheem's repetitive sequence is AGATTC. Okay, this is Chota Bheem. He will have hand mein laddu. Okay, he'll have hand mein laddu. Now, let's see. Okay, now this Chota Bheem sequence is this, but Chota Bheem has a mutated gene. Something happened and mutation has happened. Now what is going to happen? Now the gene has got mutated. Let's say, students, I am only changing. Okay, this is not practically, I don't know if such a disease will come. Instead of G, okay, instead of G, A has come. Okay, A has come. Okay, this is the mutated gene. Okay, mutated. SNP, single nucleotide polymorphism has happened. Now what will happen? I know that the repetitive sequence is AGATTC. So I will make a probe. Okay, the probe that I make for Chota Bheem. Okay, the probe that I make for Chota Bheem is A. So T, G, C, A, T, A, G. This is the probe I made. Now what will happen, students? I send this probe for autoradiography. Okay, I cannot see your charts. Am I lagging or something? Okay, yes. So here what happens, students? I have sent a probe. Okay, I have sent a probe with the repetitive sequence that I know. Now what is going to happen? The probe will not go and bind with this fellow. Why? Because this is the gene that now this fellow has. That is AATTC. But the probe that I made is not going to bind here, right? Here it is not going to bind, but it will bind here. So here what will happen, students? You will notice that, see, the mutated gene will hence not appear on the photographic film because the probe will not be complementary with the mutated gene. Clear? So it will not be complementary with the mutated gene. So since this is mutated, this fellow will not, this will bind with here. It will be not binding here. So you will not be able to see. So here we will understand that mutation has happened. Okay. So ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigen, that is protein, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen. We can do antibody or antigen also. This is used for AIDS again. Okay, AIDS it will be used. Done. Last topic we have transgenic animals. Transgenic animals is manipulated, okay? Their genes are manipulated according to our benefit. That is what is this. So animals that have their DNA manipulated or to possess, okay, DNA manipulated to possess and express an extra foreign gene, okay, an extra gene or a foreign gene is known as transgenic animals. Transgenic rats are there, rabbits are there, pigs, sheep, cows and fish have been produced, although over 95%, this is important question has come in 2005, 95% of all the existing transgenic animals are mice. That means students, we can test, okay, we can test it on chemicals, we can test it on your moisturizers, serums, all this we use mice for testing, okay? Now they have avoided maximum, they are trying to make animal, free chemicals, but still mice is the most commonly used transgenic animals, okay? Mice is the most commonly used animal. This question has come in 2004 or 5 it has come, okay? Why are these animals being produced and how can they benefit? So this beneficial question is important. Why are we using transgenic animals? First of all, normal physiology and development. That is students, transgenic animals can be specifically designed to allow, important, stay there. Yes, good boy. Okay, to allow the study of how genes are regulated, to learn about the process. Also we can use transgenic animals. Can I call all of you and tell that I want to learn something, so can I inject you and test you on you? No, right? I cannot do that. So I can take animals, I can learn about the different developments, I can understand that, okay, in this field I can use this medicines, therapeutics, I can test on them. So here you look here, regulated and how they affect the normal function of the body and its development. Study of complex factors involved in growth such as insulin, okay, by introducing genes from other species that alter the formation of this factor and studying the biological effect of the results. So first thing is to learn and understand the development, okay. Second thing, study of diseases. This is the major thing. Why did you kill 2 mice? May I know please? I killed 1 mice and you people still hold me accountable for it. Okay, I didn't kill a mice, sorry, I killed a frog. That was by mistake, students. It came and sat near my door. Okay, look here. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of diseases. These are specifically made to serve as models for human disease so that investigation of new treatment for the disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. Okay, so second thing is to study about diseases, again because we cannot test it on us, so we have to do it on animals. Okay, we have to do it on animals so that we are safe and sound. Now biological products, very important question will come from this topic. Other topics question will not come, but that 95% just remember, okay, 95% of the transgenic animals are mice. Okay, biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. So what we will do, transgenic animals that produce useful biological products can be created by introduction of a portion of a gene. That means they are already making, okay, now cow is already making milk. I want to add lactoalbumin into that. So I will just introduce that desired gene into this, right? So I will introduce a gene into it which codes for a particular product such as human protein. Students, many students make doubt. Human hormone, okay, human hormone they will make, okay. So here human protein called alpha-1 antitrypsin used to treat emphysema. Why? How does emphysema happen? Because of chain smoking, okay, because of smoking. Yes or no? Because of continuous smoking, emphysema happens. So to protect a person from emphysema, we can make a human protein alpha-1 antitrypsin. Now the catch of the question, they will give human hormone, they will give hormone, they'll give human protein, they will give something else also they can give, okay. So you have to identify. Many students will blindly write human hormone. Protein is different, hormone is different. No. In how much? 1997. The first transgenic cow called Rosie was introduced to make a human protein enriched milk. Okay, human protein enriched milk, that is 2.4 grams per liter. The milk contained human alpha lactose lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Okay, understood? So remember the date 19. Net issue, my issue. Was nutritionally a more balanced product for human beings. Now fine, no? Okay. Some problem is there. Okay, listen. Yes. Notes, you will get it on Biology by Gopika Telegram. Someone type the Telegram name in the chat box. And you will get it on Vedantu NEET English. These two telegrams, okay? These two telegrams, all the notes will be available. Clear? Yes. Okay. Okay, now it's fine, no? So students, this is most important. In 1997, Rosie was a cow to which we added a desired gene. That's a transgenic animal. That means we have manipulated the gene. We have added a foreign gene into this. What did we manipulate? Why did we manipulate? We wanted alpha lactalbumin, which was more enriched or improve in nutritional value, just like how you did golden rice, no? To improve the nutritional value for giving for babies. Okay, babies. Got it? Yes. So that is the transgenic animals. Next students, why do we use transgenic animals? For vaccine safety. Transgenic mice have been used for the use of testing the safety of a vaccine before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine. Okay, they can ask you this question. Mice is used for testing which vaccine? Polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of the batches. Okay, yes. The video part got skipped. Like, did it get stuck? Is it okay? Oh God, this lagging is too much nowadays. You people are not wanting the class too much, that's why. Okay, monkeys you heard, no? That was only the topic, okay. Chemical safety testing. This is known as toxicity or safety testing. This is done by maximum of the skin. Okay, your what do you call that, students? Skincare industry. Okay, they do the maximum. The procedure is the same as used testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substance than the non-transgenic animals. They are then exposed to toxic substance and the effects are studied. So all this you don't have to study. Okay, all this you don't have to study. Some portions only you have to study. That is like general. Just know all the steps, why they are used. First of all, to understand the normal physiology. Second thing, why we use it for biological products. Third thing is to study of diseases. Okay, and then vaccine safety and toxicity. Okay, 5 things why we use transgenic animals. What are transgenic animals? The animals whose genes are manipulated or an extra gene is added, a foreign gene is added to them so that we can get whatever we require. Okay, now one more important, very important thing. Now if you are trying to make basmati, you have to go through some processes, right? Students, 1 second. Yes. Okay. Okay, now students, see, one company got a patent for basmati rice. Okay, basmati rice, which was in India, through the US patent and the trademark office. This allowed the company to sell a new variety. Okay, one company people made basmati rice in US patent, they got off, okay, the trademark, telling that, okay, you people can sell the rice now. Okay, new variety of basmati in US and abroad. This new variety of basmati actually was derived from an Indian farmer's variety. So can I tell, okay, is it still lagging? I think it's only for sometimes, students. Okay, sometimes. Okay, now students, look here. Oh God, I have to tell these people about this issue. Okay, yes. So here what happened, Indian basmati, you can tell it in your pronunciation. Indian basmati was crossed with a semi-dwarf variety and claimed as an invention. That means I took the basmati rice, I took a small dwarf variety, I mixed this both, I got a new variety. But The actual basmati rice was already made in India by a farmer and got a patent for it. This becomes what, students? This becomes cheating, just like how we get copyright, right? So here what happened, the patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Okay, several attempts have been made to patent uses, products and processes based on Indian traditional herbal medicine. Example, turmeric, neem. What people do, no? What people do, they take turmeric, they take neem, they mix it, make a face mask and sell it in abroad, telling that this is a new variety, and they get patent for it. They get paid for it as well, because they are getting the trademark, they are getting the seal to sell this. But actually these varieties were already done by our ancestors, by our grandparents in the home. So later it becomes a problem. Okay, later it becomes a problem. So that important, what you have to study. Biopiracy is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization. Okay, without proper authorization, multinational companies and other people use resources. Okay, use resources, that is called as biopiracy, from the countries and people concerned without compensatory payment. Like you can ask, many people are there, no? These people do reaction videos, have you noticed? Many people complain for each other. They give copyright, telling you took my video, how can you take my video and do copy, like roast me. So what people do, people give copyright strikes. What is that? I have done a video. Reactors are there, roasters are there. Their job itself is to take my video and roast. Okay, yes. So what is going to happen, students? Without, like, they can ask me. If I don't agree, they're not supposed to take. I think all of you got to know, no, recently that Nayanthara Dhanush issue happened. That was similarly based on this thing only, no? So one person, the producer is telling, I do not want to give any clip of the movie. The actress is telling that I want the clip of the movie to show a small portion of how our entire story started. Now this becomes what? An issue. Same thing happens here. If a multinational company or some national companies are utilizing something from someone without their consent, without giving them any payment, it becomes biopiracy. Okay. The Indian Parliament has recently cleared the second amendment of Indian patent bills. Okay. We have just modified it. That is, take such issues into consideration, including patent terms, emergency provision and research and development. Now here what you have to remember? You have to remember second amendment, Indian Parliament. Okay, whatever words you have to remember, I am underlining. MCQ, no? Correct? I think all of you got to know that issue, no? It was very viral on my YouTube. Yeah? It was continuously popping up as if I am their cousin or something. Okay, do some few questions and after that we are done. And we are coming to the fourth class of Biology 360 Club. Now, we have to think about what next. Okay, some of you asked me for 11th portion. We will take a break from Biology 360 Club maybe. So this might be a class before the break. Okay, I will let you know, people, because next chapters, or in the comments, please drop which chapters you want from me and which chapters you want from Basava sir. Please don't tell that, ma'am, can you take biomolecules, sir, can you take molecular basis of inheritance? People, we have taught this both chapters, okay, in each other way. If you're not understanding the chapter, that means you don't know anything about the chapter. So please try to read something and then, you know, see that, okay, if you're understanding or not. Students, molecular basis of inheritance, blindly, if you learn it in one class, it will not happen. See, even if you want to learn someone's number, your favorite person's number, your parents' number, you take some time, no? Like that only these chapters are. We are not magicians. We can only teach. We can only put it across properly to you. Rest is your job. Okay, so please drop it in the comments which chapter you want from me and which chapter from Basava sir. We'll make a combination and take. Okay, tell me the answer. Very, very good. Insect pest. So +4 in the chats. Okay, GM marks is +4. NEET 2020 questions. Students, don't worry. I'll share the questions with the people and the links also will be updated. You can do 50 questions from this chapter. Match the following columns and select the correct option. BT cotton, adenosine deaminase deficiency, RNAi and PCR. Tell the answer quickly. Yes. Very good. Okay, plus marks. What's the answer here? Okay, wait. BT cotton, Bacillus thuringiensis, adenosine, gene therapy, RNA interference, cellular defense, PCR, detection of HIV. So A4, only one option is there, option number C, +8. NEET 2020 question again. Next question. RNAi is a mechanism to silence genes with the help of ssRNA, dsRNA, ssDNA, dsDNA. Yes, I can see your messages, Madhumati. Very good. dsRNA, right? dsRNA +12. Okay, +12. Next question. Golden rice is genetically modified crop plant where the incorporated gene is meant for biosynthesis of. Okay, biosynthesis of. I'll do one small change here. Yes. Sorry. Golden rice is a genetically modified crop plant where the incorporated gene is meant for the biosynthesis of. Yes, very good. Option D, that was, I think, 16. Okay, here. The first human hormone produced by recombinant DNA technology is estrogen, progesterone, thyroxine, insulin. NEET 2014 question. Yes. Very good, very good. All of you are answering. Those who joined right now, like the video as well. Okay, and don't forget to comment. Perfect. D, insulin, +20 marks. Okay, today also I have done 20 questions, 20 marks. Now students, this links will be updated. Don't take tension. Okay, I will make you solve more questions from all of the chapters because we guaranteed you that maximum marks from Biology 360. So students, here 2 links are there. No, this will be updated. Okay, this by mistake we did not check. And students, those who were asking for NEET 37 years PYQ books is still available for all of you. See, this is the price right now. Okay, it's below 1000. You will get chemistry, biology as well as physics. Okay, together you will get, and you can see the reviews are really good. 5-star reviews are there. What you have to do, just go and buy it from the Vedantu store. Okay, I think it should be available on Amazon as well. Just check on Amazon. Otherwise, you can buy it from the Vedantu store, because Vedantu store, easier thing is you can contact the support team in case the delivery does not happen or something like that. So it is at 1000 rupees, below 1000, 900 rupees right now. 37 years NEET PYQs are there. Please buy it if you have not invested on anything. Please buy and solve every day at least 50 to 60 questions. Every single day, 1 hour to 2 hours. Solve at least 100 questions. Let me tell you students, every student who is going to offline, online, everyone has a PYQ book. So I would request all of you, don't think that, okay, he is not studying, she is not studying. Please solve PYQs, because what makes you different from a 12th person? 12th person also knows NCERT. You also know NCERT. What makes you different? All this 12th people only will be writing. So your actual competition is with that 6 lakhs who has trained for NEET. So whenever some additional information is given, don't be like, why I have to study this? Please solve these, okay, and buy this book and practice. Fine? Okay, so thank you so much for joining. I hope all of you enjoyed. Yes, don't worry. Veera Babu, the stream will take some time because it's a long stream. No, it will take some time. It will refresh. Don't worry, the class will be updated in 2 hours. You will be able to see Baswa sir's class as well. So I want all of you to comment and let me know how Biology 360 Club was. So thank you so much for joining. See you all in next class. And I hope all of you enjoyed this class. Please do let us know in the comments how the class was, and like the video if you're watching it later. If you have any doubts, drop it in the comments. And most important comment I want is which chapter you want from us next. Otherwise, we will keep a halt for Biology 360 Club, and then we will go into the 11th portion. Okay, so thank you so much students. See you all. Take care. Go have dinner and have a nice sleep.